you people will be asking questions, and I hope you people will ask questions in this session. So without further ado, let me start with myself. You people know my name, most of you in fact know, and about the first 2-3 rows, those who don't know, I'm Ahmad Afzal Kiani. I'm associate professor and also a program manager of BS Biosciences and BS Biotechnology program. I did my PhD in 2014 from University of Karachi. So by training, by education, I'm biotechnologist. My core area of specialization is agriculture biotechnology, agriculture plant biotechnology. So that's a brief about myself. Now I would request one by one each of the panelists that they should tell their experience and their background with us. So we'll start with Dr. Saeed Khan.
Thank you very much, Ahmad. मैं तो समझा 1 घंटा है, आधा घंटा आपने ले लिया तो हम सलाम दुआ करके निकल जाएंगे. टाइम तो ख़त्म हो गया, इतना डिस्कशन के लिए तो कम टाइम रह गया. Thank you, Ahmad. It was pleasure to be here, and thank you the organizer for inviting us. I think life science is a vast area, and once a graduate enter into a school for particular field, these concerns are in their mind that what would be the outcome of the selection of the subject, it is what would be the career outcome. So everyone has this in mind, but life sciences, the good thing is life sciences, there's a diverse field. And in Pakistan, though we have limited opportunities, but for the life sciences, the maximum are, I think, have for the life science graduates. I will share my experience. I did my master in microbiology, so is a PhD in microbiology from University of Karachi in collaboration with Aga Khan University. Then I did my postdoc from University of Washington, Seattle. And then I joined Aga Khan Dow University in 2011 as assistant professor, been promoted as associate in 2015 and become full professor in 2018. So I was lucky to have the career very quick, and though I was not, things were not that planned, but I'm thankful to Almighty Allah. And again to the life sciences that the field was quite fertile for me, and in terms of the opportunity that I got. And the performance also is a matter, because once the timing very much important. When you get an opportunity and you perform, so getting opportunities, I think you can create opportunity as well. You may be lucky to have an opportunity, but then you create opportunity. And the current era of the pandemic, again, is very much for the life science graduates. The performance done by the life science graduates in this pandemic is very much obvious from all aspects, right, from research, diagnostics, and helping the infected person or diagnosing the COVID in diagnostic labs as well, doing research. So this is what is life science graduate has can perform, and there are different areas then you can perform in research, diagnostics, pharmaceuticals, teaching. It's a vast area. Then there are some other direction that you will be like witness that sometimes we are going in narrow tunnel. If you think out of the box, there are many ways that you can choose from. The good thing, when you have a selection, you are a lucky person. When you are forced to do something, even that is something very high-fi, but if you are being forced to do, then you cannot enjoy that. If you choose from out of many things, so you will enjoy and you can perform better. So the other areas that my experience that I did, like ASM, American Society for Microbiology, I'm currently the president of Pakistan Biological Safety Association and ambassador to Pakistan by American Society for Microbiology. So these are other areas, the life sciences, that you can interact with people. The good asset is interaction. You all in this session not just listening us, but you are interacting, engaging other context and creating context. So we will be sharing other thing because Hamad is looking at me and he's looking at his watch to take revenge. So I will hand over the mic to Aftab and we will continue discussion in another session. Thank you.
Thank you, Dr. Saeed. It's indeed great pleasure for me to be here at SZABIST. I used to come here like previously as well. Thank you very much for your kind invitation. I traveled from Islamabad to join this discussion. And I think it's very hard actually after the lunch because after the lunch, the circulation between intestine and liver that increased and the circulation from your heart to brain decreased. So it's very hard to actually understand the things, but we'll try that you keep, you stay wake up during our discussion. You are not sleepy anymore. So as Dr. Saeedi has mentioned that there are a lot of things that we can do being the science graduates. My journey is like I'm graduate from Punjab University. I did my graduation in microbiology and molecular genetics. And then my MPhil is in biotechnology. My PhD is in stem cell biology. And my postdoc is in tissue engineering. So I moved quite a diverse field, starting from microbiology, ending with tissue engineering. I was working in the United States back in 2008. I had the opportunity to work in an industry as well. So I had an experience of working in industry in US. And there I thought that there is actually need of having a platform of our young researchers in Pakistan because the opportunities for young researchers are like few in Pakistan. And resources we also have, we don't have a lot of resources in our country. So like thinking there in US, how we can improve science and technology in Pakistan. When I moved back in 2009, we started National Academy of Young Scientists. I think most of you know about NAIS Pakistan. So we started back in 2009 and so far we are now lucky to be connected with around 50,000 young researchers Pakistan who are working in Pakistan and as well as the researchers who are working in different countries of the world. So this is another journey I took like back in 2009 we started NAIS and it's almost 12 years now. And beside that like I was from the start of NAIS, we used to join different meetings on academia industry linkages, but there is a lot more discussion rather the concrete steps. So it's always like there is a gap between academia on one side and industry on the other side. So I was also part of HEC program, so I used to review the projects, they were related to academia and industry. So there I thought that it's a gap, especially in the life sciences field. So how we can fill this gap? So being like not joining the university as a part of like assistant professor, I started my own company back in 2018. That's Chattha Biocare. It's based, the office is in NUST, in First Science and Technology Park in Islamabad. So we are working quite a diverse projects and I'll share some of the examples that what path we are taking. Beside that, as Dr. Saeed mentioned that he's a part of American Society for Microbiology. I also had the opportunity to be part of World Economic Forum as a scientist member of World Economic Forum and World Science Forum. So these are the bigger forums at international level. And in all these forums, we also used to discuss the challenges, like the things we are discussing now. What are the challenges to our young researchers? What are the opportunities for the young researchers? What are the global issues and how young researchers can solve those issues? So these kind of discussion we used to do. And I'm really happy that they have included this kind of like panel discussion in this program. And I'm quite hopeful that it will be fruitful for all of you. So with this, I hand over to.
Thank you so much. Jubair is here. Thank you for kind invitations. Basically, I am a chemist and concerned with the pharmaceutical companies manufacturing. Basically, we manufacture life-saving drugs. So life science is a very vast field, and I am operating as a quality control head in pharmaceutical manufacturing. We manufacture life-saving drugs. So thank you so much for invitation.
All right, thank you all three of you, and obviously you people are all from very diverse background. As Dr. Saeed was mentioning कि मैंने काफ़ी time ले लिया, about half an hour. Well, Dr. Saeed is used to getting a lot of attention because during the pandemic he was often seen on the electronic media because he was the spokesperson and he has a very good knowledge about this whole COVID phenomena and I can relate to him as well because I'm the member of Pakistan Biological Safety Association, for which he is presiding right now. So moving on to the grave part of this academics that we are facing right now, as in the challenges that we are facing right now. Every life sciences graduate, potential life science graduate that is sitting here must be thinking that what next for me after the degree. And while you people were telling about your journey, your beautiful journey, I've noticed that you people are involved in this field for no less than 15 years. Dr. Saeed mentioned, Dr. Aftab mentioned, and Zubair also mentioned this. So I would like to know that what are the challenges that you have seen while you have reached here, that you as a graduate, as a potential, as a student faced, and now you are seeing that these graduates that are coming into the market are facing right now. So I would like to each and every one of you comment. So again, we'll start with Dr. Saeed, please. Yes, we can surely, no matter.
No, I will not allow this. Okay, the main challenge is that the graduate is usually not ready for the market. This is the main challenge, that once you go through all the books and when you come into the market, you are just blank. I will share my experience when I joined SIUT, Indian Institute of Urology, for my master. So we read a lot about DNA and things like that, and RNA, protein. But when we were exposed to the bench, extract DNA, we are just hands up. How to do extract DNA, how it looks like. So this is the main issue, that usually when we're studying in the course or the syllabi that we have, they are focused more on books rather more on practical things. So that is the missing thing. And then we have like too many graduates all together. How they will find, we have very limited routine opportunities. Like if a science graduate from microbiology, usually he or she will be looking for any of the position in the clinical lab or in pharma. So how could it possible to arrange thousands of positions all together for too many institutes? So usually we need to like work on this, that you need to be the one. If there's one opportunity, you should be the one to avail that opportunity. You need to be capable for that. And our area, that was nowadays is very easy to have access like internship program to be prepared for the market. We usually not aware of that thing in our time, that internship. There were hardly any opportunities available. Usually the labs or the industry, they don't have connection with the institution like university or the colleges. So that was lacking in our time. But now in my lab, I have like every day internships. Interns come to my lab and they're asking for internship, and I just randomly allow them just for a week, for a month, whatever time they have. Allow them to they see the equipments and they feel the reality of the life sciences molecule, like RNA, DNA. They should touch the pipettes and they should be aware of the environment. So this is what I will recommend, that though the positions are very much limited, but each and every one of you should be capable to be able to avail the opportunity. And you should have some extra qualification along with the routine degree with you. Like if you are just doing a degree from here, and someone else has extra capability, writing skills, or someone has communication skills. So these can make you prominent during the interview. And if there's only single opportunity and you have extra quality or qualification, then you can avail that. So I will encourage that each and every graduate should try for internship during the graduation, before you get your degree. Whenever you have these summer vacation or whatever gaps you have in the middle, no matter it's a week long or month long, you should go for it. So you should be prepared before time. When you are graduated and you going black in the interview, and then 2-3 सवाल हम करते हैं, पता चलता है वो blank है, तो फिर हालांकि वो टाइम नहीं होता कि उस पर आप डिसाइड करें, लेकिन बेटर कैंडिडेट अवेलेबल होता है तो हम उसको चूज़ कर लेते हैं. सो ये इंटर्नशिप बहुत आपको इनकरेज करता है और आपकी कॉन्फिडेंस को बिल्ड करता है, तो आप ये इंश्योर कर लें कि आप भी अपने प्रोग्राम में ये कंपलसरी कर लें और कनेक्शन बनाएं कि मेक इट ईजियर फॉर योर ग्रेजुएट टू डू इंटर्नशिप बिफोर द कंप्लीशन ऑफ द डिग्री.
बहुत इम्पॉर्टेंट पॉइंट रेज किया डॉक्टर सईद ने, बट आई वुड लाइक टू टेल यू that we do have internship program included. In fact, it's a mandatory part for graduation. Each and every student must go for internship for at least 40 days, 6 weeks. So ये part हमारे academics का, और since the inception of this program, since 2011, this is the part of our curriculum. Exactly. But I have a different question related to that. I'll ask that later. I would like Dr. Aftab to share his views about the challenges.
Well, I had the opportunity to travel to different countries. Dr. Saeed, he's also traveling a lot. But I traveled to like very developed countries and also the developing countries. I love to go to different universities, interacting with the students and faculty. In Pakistan, I think I've been to around 100 universities of Pakistan and interacting with the faculty members and students. So there are two, like, for the challenges, I must say there are like two parts. One is on the students end and one is on the university end or the faculty end. So if we talk about the student part, so let's ask you a question. How many of you want to do job after your graduation? And how many of you would like to have their own company or startup? So almost 50-50. So it's a good thing that things are changing now, because before this, like almost 90% they wanted to have a job, and very few they wanted to have their own startup or their own company. So for the students end, you should go for cash. Cash is very important. Is it important? So cash is, I must say, cash is K for knowledge, A is for attitude, S is for skills, and H is for habits. So this cash is actually very important for you. In universities, we mostly go for like knowledge. If we compare the knowledge being delivered in Pakistan, that's almost the same being delivered in almost all the foreign universities, like in most of the developed countries. The knowledge is same, the books we read are the same. The two things, like if we talk about the skills, we are like to some extent we go for the same skills. Like if you are going for DNA extraction, in some of the universities we have very good facilities for DNA extraction, protein analysis, or the biochemistry, or the microbiology, these kind of things. So you have some kind of experience of that. There are two things which are actually mainly lacking in Pakistan. That is your attitude and the habits. This is very important to actually develop during your university days. So if you develop very good habits, actually it will take a whole lecture actually if we talk about the habits, but you have the idea being the graduates that what are the habits, what is your attitude. So attitude meant a lot. Like, you know, I'm in NURST, for example, and I had the interaction with a student from NURST. They have a different attitude. Being like, whenever they are at some forum, they have a different attitude. That's being developed in the universities. If you are going, like Qaeda University is top ranked university in Pakistan, but the Qaeda University students' attitude is different. The university, like the students from NURST, the attitude is different. Student from Punjab University, the attitude is different. Sindh University, Jamshoro student attitude is different. So this is actually very important that you build a good attitude while you are in the universities, and also your habits. So these mental are, so you should focus on that. Another very important thing from the students end is, like, I must say that, and you should remember this for the whole life, 70% of your success in your future life, either you are going for some job, or you are going to have some startups, or you are going to start some company. 70% of your success is dependent on your network. And universities are the best places to develop the network. Your friend circle is very important to have a good networking with the faculty members. Like the interaction, we are here today, a professor from UCL, he's here today. So you should interact with these people. Professor Saeed, he has like a very good, so if you are connected to this guy, this person, you are connected to whole Pakistan and rest of the world. So this is how you actually develop the connections, and these connections help you a lot in your future life. So this is the end, the students' end. On the university end, what I have seen the challenge in Pakistan university, one of the thing is lack of infrastructure. So I was talking to a few faculty members yesterday when I arrived to Karachi University, and they were complaining that we lack some equipments, like even we don't have the PCR machines, sometimes we have to run the HPLC, we don't have that much facilities. So one of the challenges is the infrastructure. We don't have that much infrastructure, but this is not the big challenge. The big challenge in Pakistan from the university's end is the freedom. Like I talked with one of the Nobel laureate, Professor Tim Hunt. He is from the Cambridge University. So I asked him that how Cambridge University has produced almost 100 Nobel laureates, and the whole Muslim world, they have produced just 4 Nobel laureates, and even those they have worked in the foreign universities. So what is the difference? And he said one of the key elements that we provide in our universities is the freedom of work. Like each student, they have a different in their mind. Somebody want to work on virology, somebody want to work in immunology, someone is interested in microbiology. So each person has a different interest. Even in, for example, microbiology, they have a very diverse interest. So in Pakistan, what I have after talking to so many faculty members, unfortunately, they complain about the faculty members that they don't give them that much freedom. If they want to do the experiment, for example, in certain field, this is what I also during my own journey. I wanted to do something else, but the professor says that we don't have those much facilities or expertise, so you have to confine yourself what we are doing. When you have passion for something, when you have really a deep passion from inside, like that actually build up, like somebody is suffering from some disease, infected disease or non-communicable disease in your family. For example, somebody is suffering from diabetes, and you really want to cure the diabetes in your graduation, and you have a lot of energy to do that, but you want to work on that area, but your supervisor, he want you to work in area microbiology, for example, then you lose the interest. So I think the universities, they should provide more freedom to the students and should provide more opportunity. Another point Dr. Saeed raised is very important for the researchers, like the internship opportunity. You should go for that. During your free time, you should utilize that time in whatever skills you can opt. Skills are very important, and you should also focus on the future skill. And you should know what are the future skills. You know, everything is changing very rapidly. Like the things were very different back in 2000, and then they were very much different in 2010. The things are very much changed to 2020. The things will be totally changed in 2030. So you have to upgrade yourself. Just like we have a software version like software version 1.0, then we go up in 1.5, then to 2, 2.5, 3, and so on. This is how we upgrade the software. For example, even the software for DNA analysis, that's being upgraded. You should also upgrade yourself. This is your responsibility, and when you upgrade yourself according to the need of the hour, and also thinking about the future, what challenges we are going to face in the future, what skills you said you have, what is the skill set now you have to work for the 2025, you should keep that in mind. And if you will prepare yourself for that, definitely you can do a much better job. So there is a lot of things, but I think time is short, so I must restrict myself here. And
राइट जो ऐसा मेरा आपसे एक थोड़ा डिफरेंट क्वेश्चन होगा, क्योंकि बेसिकली यू आर फ्रॉम फार्मास्यूटिकल सेक्टर. तो आप स्टूडेंट जब ग्रेजुएट होते हैं, ऑब्वियसली वो अप्लाई कर रहे होते हैं इन फार्मास्यूटिकल सेक्टर, एंड यू रिप्रेजेंट फार्मास्यूटिकल सेक्टर हियर, यू आर पार्ट ऑफ दैट. कौन से स्किल सेट्स हैं जो आपको चाहिए होते हैं एंड यू डोंट फाइंड इन द स्टूडेंट्स, इन द ग्रेजुएट्स, फॉर विच यू बेसिकली रिफ्यूज देम और आस्क देम टू इम्प्रूव अपॉन?
बेसिकली जब कोई जॉब के लिए अप्लाई करते हैं, ख़ासतौर पर फार्मास्यूटिकल इंडस्ट्रीज़ में, तो उसकी सीवी जो है, रिज्यूम इम्पॉर्टेंट है, उसको सॉर्ट आउट किया जाता है एचआर डिपार्टमेंट में. अगर आपकी सीवी या रिज्यूम अच्छा है तो फिर उसको सॉर्ट आउट करके इंटरव्यू के लिए बुलाया जाता है. इंटरव्यू में आपका डिपेंड करता है आपकी स्किल्स, आपका कम्युनिकेशंस, आपका कॉन्फिडेंस, और थियोरिटिकली जो आपने साइंस ग्रेजुएट किया है, मिसाल केमिस्ट हैं, आप माइक्रोबायोलॉजिस्ट हैं, तो उसमें थियोरिटिकली भी स्ट्रांग होना चाहिए. आप उसमें जो इंटरव्यूज़ कलेक्ट करने वाला है वो उसको आपका कॉन्फिडेंस, आपके स्किल्स दोनों देखते हैं. खुद इंटरव्यूज़ करते हैं तो उसमें ये चीज़ें ज़रूर देखी जाती है, उसमें हम सॉर्ट आउट करते हैं स्टूडेंट्स को और उसको अपॉइंट करते हैं।
ऑल राइट, डॉक्टर सईद, एक क्वेश्चन मेरा स्पेसिफिकली आपसे होगा कि बिकॉज यू हैव हेल्ड सो मैनी लीडरशिप रोल्स, सो और एक लैब को भी आप हेड कर रहे हैं, तो हमारे स्टूडेंट्स जब ग्रेजुएट होते हैं, और यू हैव मेंशन अबाउट इंटर्नशिप, डॉक्टर आफ्ताब हैज़ आल्सो मेंशनड इट, students or most of the universities are offering internships right now. In fact, the universities I know and people are sitting here who are running these programs in their universities as well. They are also running these internship programs. But kuch universities ki graduates based on their internship alone are eligible or sort of qualify for that job. और बहुत ज़्यादा केसेस में ऐसे होता है कि स्टूडेंट इंटर्नशिप कर चुका होता है एटलीस्ट 6 वीक्स की, स्टिल एम्प्लॉयर उनको रिफ्यूज़ करता है, कोई लैब का इंचार्ज रिफ्यूज़ करेगा, कोई फार्मास्यूटिकल का इंचार्ज, कोई एम्प्लॉयर रिफ्यूज़ करेगा. सो व्हाई इज़ दैट? आई मीन क्या स्किल सेट्स चाहिए होते हैं स्टिल उस ग्रेजुएट को जो कि इंटर्नशिप के बावजूद भी वो अचीव नहीं कर पा रहा है?
बहुत अहम क्वेश्चन किया है. एक तो मेन चीज़ होती है कि अपॉर्चुनिटीज़ कितनी है. कभी कभी ये होता है ना कि उसको रिफ्यूज़ करना ही होता है, तो अनफॉर्चुनेटली ये चीज़ होती है कि उन्होंने रिफ्यूज़ करना है और या उसके पास एक obvious best choice available है, so the employer usually go for it. Important चीज़ ये है कि इसका mentorship बहुत important है. Usually students इस चीज़ को सिर्फ डिग्री लेके निकल जाते हैं और कोई बाकायदा काउंसलिंग अपने टीचर से या ऐसे बंदे से नहीं करते शुरू में जो उनको वो चीज़ें बता दे जो अभी मैं अगर मैं बता रहा हूँ, तो ये इंटरव्यू में जाने से पहले वो अपने मेंटोर से ये चीज़ शेयर कर लें. इस तरह हमारे अपने स्टूडेंट्स होते हैं, स्टूडेंट्स ग्रेजुएट करते हैं, उनको हम काउंसलिंग बाकायदा कर रहे होते हैं और उनसे हमारी कनेक्शन होती है कि भाई जिस तरह सर ने अभी बताया कि रिज्यूमे किस तरह बनाया जाता है ना, आपकी रिज्यूमे का ऑर्डर ही सही नहीं है, तो हमें तो ऐसी कोई चीज़ नुक्ता चाहिए कि जिसे रिजेक्ट कर लें, क्योंकि नंबर्स कैंडिडेट के बहुत सारे हैं और एक बंदा या 2 बंदे लेने हैं. तो उसमें अगर आपके पास मेंटोरशिप, आपने किसी मेंटोर को अच्छा कंसल्ट किया और आपका मेंटोर अच्छा है और उन्होंने आपको गाइड किया है, तो CV आपकी बहुत अच्छी होनी चाहिए. कभी कभी हम अपने आप को अच्छा प्रेजेंट नहीं करें, हमारे में क्वालिटी होती है बट वी डोंट प्रेजेंट प्रॉपरली और सेल्फ. सो वी नीड टू फर्स्ट वो ये है कि आपका रिज्यूमे बिल्कुल अच्छे ऑर्डर में हो. देन कम्युनिकेशन स्किल्स के बिल्कुल आपने वो कंपटीशन जीतना है. अगर कंपटीशन है 5-10 लोगों का एक पोजीशन के लिए, तो कम्युनिकेशन स्किल्स उसी पर्टिकुलर टाइम में आपने एम्प्लॉयर को जो ना कन्विंस करना है कि यू आर द राइट पर्सन. सो यूजुअली कम्युनिकेशन स्किल्स, लोग अक्सर आते बहुत खौफ में होते हैं, कैंडिडेट जब भी आते हैं बेचारा बड़ा सहमा होता है, पता नहीं कोई क़यामत वाली घड़ी है. तो ये कम्युनिकेशन स्किल्स बहुत इंपॉर्टेंट होता है. कभी कभार अक्सर ये होता है कि वो बंदा उस वक्त फेल होता है, लेकिन हमारी नज़र होती है कि भाई ये बंदा पोटेंशियल इसमें अच्छा है, लेकिन वो हमें बहुत अच्छा बनना पड़ता है कि इस पर हम इतना नज़र रखें कि भाई ये इस वक्त तो परफॉर्मेंस इसकी सही नहीं है, लेकिन बंदे में पोटेंशियल है, तो उसको उस वक्त परफॉर्म करना चाहिए कि उसकी कम्युनिकेशन स्किल्स बहुत अच्छी होनी चाहिए. और मार्केट रिलेटेड या जॉब रिलेटेड उसकी जितनी भी एक्सपीरियंस है वो उस वक्त प्रेजेंट करें. लाइक एंटीबायोटिक और माइक्रोबायोलॉजी रिलेटेड 2-3 हम क्वेश्चन अक्सर कर लेते हैं कि भाई आप नाम बताएं बैक्टीरिया का, सिंपल का है, हमें आइडिया हो जाए. तो अक्सर स्टूडेंट्स कहता है भाई मैं तो यहाँ से भी काफ़ी स्टूडेंट्स आए कि भाई मैं तो तैयार नहीं था इंटरव्यू के लिए, मैं तो वैसे आ गया था और ऐसे आ गई थी, तो मेरे अभी मेरे फेवरेट क्वेश्चन है, तो बिलाल अभी बैठा हुआ है तो मैं कह 2 ग्राम पॉजिटिव बताओ 2 ग्राम नेगेटिव बताओ, जस्ट टू हैव अ सेंस. तो अक्सर वो इतना होता है कि वो रेडी नहीं होते. सो कम्युनिकेशन स्किल, CV और कॉन्फिडेंस आपका, और मेंटोर से कम आपने किसी अपने टीचर से कंसल्ट लाजमी करना है. अगर आप जॉब के लिए जा रहे हैं, सो यू नीड टू बी प्रिपेयर्ड. अगर आप ऐसे राह चलते हुए आम अल्फाज में कहते हैं मुँह उठा के चलेंगे तो फिर आपको जॉब नहीं मिलेगी. तो प्रिपेयरनेस इज़ इम्पॉर्टेंट. और कॉन्टेक्स्ट बहुत इम्पॉर्टेंट है. दूसरी एक चीज़ मैं आपको शेयर करूँ, स्टूडेंट्स अप्लाई नहीं करते हैं, मतलब वो फेलियर पे ज़्यादा फोकस करते हैं कि मैं जॉब नहीं मिली, लेकिन उन्हें अप्लाई कितना किया होता है. अक्सर मैं स्टूडेंट्स को कहता हूँ कि आपको मतलब बाहर अगर कोई स्कॉलरशिप अप्लाई करनी है तो मैंने उनको बोला था कि आप 100 एप्लीकेशन आप डालें, अगर आपको नहीं होता तब मुझे आप बोले कि आपका नहीं हुआ. इफ़ यू अप्लाई जस्ट वंस और आप नहीं हुआ, आप रोना धोना शुरू कर दिया, तो ये इट्स नॉट द राइट थिंग. फिर इस तरह ट्रेवल ग्रांट्स है, आउटसाइड एक्सपीरियंस है, कॉन्फ्रेंसेज होती हैं. अभी ASM की कॉन्फ्रेंस हो रही है, अमेरिकन सोसाइटी फॉर माइक्रोबायोलॉजी की. मेरा अपना पर्सनल एक्सपीरियंस इससे बहुत अच्छा है. व्हेन आई स्टार्टेड तकरीबन 20-30 कंट्रीज में मैंने ट्रैवल किया है, यूजुअली ऑल थ्रू दिस स्कॉलरशिप. ट्रैवल ग्रांट्स, ASM पर्टिकुलरली वो ट्रैवल ग्रांट्स आपको देती है. सो वंस आपका एक्सपोजर हो गया प्रैक्टिकली, मतलब अक्सर मैं स्टूडेंट्स, मेरे सारे स्टूडेंट्स जो है यूजुअली दे ट्रैवल अलॉट. मैं और कोई आप प्रेजेंट करें ना करें, आप जिस जहाज़ में बैठे वहाँ ट्रैवल करें, एक्सपीरियंस करें, दैट इज़ अ लॉट ऑफ आपका कॉन्फिडेंस बिल्ड होता है. सो यूजुअली और स्टूडेंट्स देयर परफॉर्म, कुछ तो अच्छे परफॉर्म करते हैं, लेकिन सम टाइम मेरा एक्सपीरियंस जब मेरे सामने आते हैं तो वो कंफ्यूज होते हैं और सवाल का जवाब नहीं दे पाते हैं. और दैट इज़ नॉट द राइट थिंग कि उस पर हम जज करें, लेकिन वही टाइम होता है परफॉर्मेंस का. जब आपका इंटरव्यू लिया जाता है तो उस वक्त आपने परफॉर्म अच्छा करना होता है. ये बहुत सारी चीज़ें कलेक्टिवली इसमें आपको हेल्प कर सकती हैं कि आपने एक्सपोजर आपका अच्छा हो, कम्युनिकेशन स्किल्स अच्छी हो, नॉलेज उस वक्त अवेलेबल हो. अक्सर चीज़ें बाद में याद आती हैं लेकिन उसका तो फ़ायदा नहीं होता. So that what I will suggest.
Dr. Aftab, since you have mentioned about the concept of cash, so I will in fact put you on spot right now. So what matters most, attitude or skill?
Well, this is a very important question. Actually, cash is, cash is important. So in cash, each thing is equally important. So it's actually very much depends on where you are going for the job. So at some places they are looking for a job that is like mainly to deal with the thing, deal with the public. So that's not very much technical. In that case, you definitely go for the attitude of the person and the habits of the person. So in that case, that is important. But if you are going to join some laboratory, for example, if you are going to join Dr. Saeed laboratory and you have to perform some key experiments there, so in that case, your skill set is very important. For example, I am looking for a person, I am working in area of regenerative medicine or the stem cell biology. So if I have to I have a job in my place and I'm looking for a person who has to prepare the cell lines, for example, then I will definitely look for a person who has expertise in cell culture. So he must know that how he has to handle the cell lines to avoid the contamination and these kind of, so the person with that kind of expertise, that's important for me. But if I am looking for something like if I have to hire a person and he has to do like I have to buy some equipments and I have to go for distribution of that equipments. So in that person, in that kind of skill, he need to be good in communication and he need to have a good attitude. So it actually very much depends. But as he mentioned that cash is important, so for each you should dwell the similar things, like you should go for a good knowledge, you should have a better understanding. And as Dr. Seedhi was mentioning that you should actually go for, like if you are preparing a good resume or CV, that's very important for you, that it should be properly made. And you should develop it actually gradually with whenever you are like even in the second semester or in the third semester, you should start working on that and you can edit it. And then you should tailor it according to the requirements. You should know what job is going to be offered for you. So you should know that if Dr. Saeed is going to offer a job in the laboratory and you have to do some practical skill, then you should focus on that part, that you have the relevant skill sets with you. So you should tailor, that's very important actually. So we mentioned so many things in our CV. For example, my CV, if I have to send to somebody, is like 40 pages and nobody will look at that what I have done, like traveling and conferences and so on. But if I have to apply in a university, then I should mention specifically what publication I have done, what contribution I have done in like writing some chapters, what grants I have won so far and so on. So this is how you tailor the things, and this tailoring is very important for your future.
Right, thank you, Dr. Aftab. So what I have got as an impression that it is a mixture of both. I mean, you need to have skill, but you also need to have a good attitude. It's about sector in which you are applying. कहीं कुछ predominantly ज़्यादा होगा. अच्छा, डॉक्टर सईद की ख्वाहिश भी थी कि सीक्वेंस चेंज किया जाए, तो हम सीक्वेंस को भी थोड़ा सा चेंज करते हैं. एंड डिस्कसिंग अबाउट द अपॉर्चुनिटीज़ नाउ. सो जुबैर साहब, आप क्या फील करते हैं कि फार्मास्यूटिकल सेक्टर में किस किस्म की अपॉर्चुनिटीज़ अभी अवेलेबल हैं, कंसिडरिंग कि वी हैव रिसेंटली गॉन थ्रू अ वेरी बैड फेज़. एम्प्लॉयबिलिटी बहुत ज़्यादा चैलेंज हुई है, हैम्पर हुई है, कोविड का जो फेज़ हमने गुजारा है. सो अपॉर्चुनिटीज़ क्या देखते हैं फ्रेश हमारे जो ग्रेजुएट्स हैं लाइफ साइंसेस के, उनके लिए?
थैंक यू. बेसिकली फार्मास्यूटिकल में जो साइंस ग्रेजुएट हैं वो एक फार्मासिस्ट, केमिस्ट और माइक्रोबायोलॉजिस्ट, इन तीन साइंस ग्रेजुएट की अपॉर्चुनिटीज़ हैं, जिसमें फार्मासिस्ट बेसिकली प्रोडक्शन डिपार्टमेंट्स को डील करते हैं, और जो केमिस्ट हैं वो क्वालिटी कंट्रोल लेब्स को डील करते हैं। एनालिसिस हैं, इक्विपमेंट हैं, इंस्ट्रूमेंट हैं, जो भी ड्रग वहाँ बनती हैं उसको मैन्युफैक्चरिंग टेस्ट करते हैं, एनालाइज करते हैं, आइडेंटिफाई करते हैं थ्रू डिफरेंट इंस्ट्रूमेंट इक्विपमेंट के। और माइक्रोबायोलॉजिस्ट, हमारे पास एक माइक्रोबायोलॉजी सेक्शन है क्वालिटी कंट्रोल लैब में, तो वहाँ माइक्रोबायोलॉजिस्ट उसकी ड्रग की एनालिसिस करते हैं। जो भी ड्रग बनती हैं, जो किसी भी फॉर्म में हैं, टैबलेट हैं, सिरप हैं, कैप्सूल हैं, इंजेक्शन हैं, उसकी माइक्रोबायोलॉजिकल टेस्ट भी होती हैं। तो ये साइंस के 3 ग्रेजुएट हैं, जिसमें फार्मासिस्ट, केमिस्ट और माइक्रोबायोलॉजिस्ट। इनकी अपॉर्चुनिटीज़ फार्मा के अंदर बहुत अच्छी हैं और हम उनको अपॉर्चुनिटी देते हैं इसमें।
थैंक यू डॉक्टर आफ्ताब, वेरी क्विकली जी प्लीज़ आप भी अपॉर्चुनिटीज़ के हवाले से शेयर कीजिएगा, बिकॉज यू आर ऑल्सो एडिंग नेज, तो आपका एक बड़ा डाइवर्स एक्सपीरियंस होगा इन रिगार्ड्स टू दैट।
Well, I think if there is an opportunity, go for it. For like if you have done some graduation in microbiology or molecular biology, I must say that if you have don't need any sport, you don't have to support your family, then you should go for your further studies. Try to get some scholarship. And for scholarship, you should also prepare your mind. So it's very important initially that you should prepare your mind what you are going to do. Either you are going to do some job after your BS programs or MS program, or you are going to get some scholarship, and either you are going to study in Pakistan or you are going to study abroad. So first of all, it's very important actually to prepare in your mind what you are going to do, and then you prepare yourself accordingly. Either you are going to get some scholarships, so what are the requirement of scholarship? So you check which country you are going to do. Either you are going to do a PhD in US, or you are going to do PhD in UK or in European countries, in China, Malaysia, Korea, Australia, where you want to go. Because each country has a different requirement. If you want to go to US, for example, there is a requirement of GRE, so you have to clear that. In most of the countries, there is a requirement that you should have an English language course. So you should do that beforehand. So you should initially, it's actually very important for yourself. In your mind, you have to prepare yourself what you are going to do. So this is one path. Either you are going for higher studies, or you are going to do some job. For job, then you also prepare your mind. Either you are going in academic side, or you going to teach in some college, in university. That's one segment. Are you going to do some research job? For example, you are going to some in food diagnostic laboratory. Are you going to in forensic laboratory, for example? If you want to go in some other research phase. So that's another thing. Are you going to join some hospital laboratory? You are going to do the molecular biology testing, some kind of thing. So that's one side is the government side. Another target is the private sector. It's are you going to go in some industry, for example? So you have to prepare yourself. Which industry you are more interested? So it's actually, for me, it's very important for you people to prepare in your mind. Actually, the Pakistani graduates, they are very much confused. On one side, they want to go for after MPhil, they want to do PhD on one side. On another side, they want to prepare for CSS. Another side, they want to prepare for PCS. Another side, they want to apply for a teacher job. Another side, they want to experience in the hospital. And on the other side, they want to do marriage. Yes. So you should prepare yourself. It should be very clear in your mind what you are going to achieve, what you want to do, and you should prepare yourself accordingly. That's actually one segment. In most of the developed countries, if you talk about, for example, in US, in European countries, even now in China, almost 50% graduates, they go for industry. Almost half of the graduates, they go to industry and they support the industry. They do the research and development. Unfortunately, I was before this panel discussion, we were discussing that how many pharmaceutical units are there in Pakistan. That's a huge. We have more than 600 pharmaceutical units in Pakistan, but how many of those are involved in research and development? So Pakistan industry is mainly dependent. They just import the things, they pack it in Pakistan, and then they sell it. There is actually lack of research and development, and this is the area we have to fill. And this is how we have to also involve the policymakers, that they should compel the industry that they should have research and development segments, so we can engage more and more researchers. So that's another segment. So on one side, government jobs, private sector jobs. Are you going to some industry? Another very important area the whole world is focusing is the startups. You know, startups very important. Like if you looking for some problem, for example, there was a lecture, very good lecture by professor from UCL. He mentioned that there is a like growing problem of antibiotic resistance and how we can actually solve this problem. So in Pakistan, like I would like to have, like in future we can have a session on that to identify the problems. So on one side we can identify the problems and then we can have a startup to fix that problem. And there is lot in Pakistan. There are problems everywhere. So the very important thing for the graduate is to identify the problems and know your own strengths, and then you can work on that problem. And there is lot of more opportunities coming up for you. For example, I'm based in Pakistan first Science and Technology Park that was built in Islamabad in NUST. Each university is going for that. Qaeda University, they have started the Technology Science Park. There is another university that is PFIST, that is in Haripur. They have established the Technology Science Park. So mainly, so many universities, they are going for Technology Science Park. In addition, for you people, you will be aware that there are NIC, National Incubation Centers. They are in Karachi, in Lahore, all the major cities. So you can have a place in there. They provide you free space. They provide you coaching. So if you want to start something, some industry, I will give you one or two examples here. One of the problem for our female doctors was they were not in the job. More than like 50% after doing their MBBS, they were sitting at home. You know, Sehat Kahani came and they have like now recently they got the funding of $10 million. So they are providing opportunities to so many female doctors. So that was a problem, they have solved it. Another graduate, he was from Comsat University in Islamabad. He did MPhil in bioinformatics. And for that, he started his own company named as Xilexa. And in just 3 years, they are providing services to more than 20 countries. So Xilexa is expanding. So what they do, they are based on AI-based image analysis. So they have like the image analysis for the breast cancer, and they can tell with very much accuracy that the tumor is benign or is malignant or there is no tumor and so on. So this is how they are providing the services even to the developed countries. So this is how that was an area in which they actually worked, and now they have employed, I think, more than 20 researchers who are working in that company. Even in my company, I'm working on biofertilizers on one side, I'm working on probiotics and some of the products related to regenerative medicine. For each segments, we are running the trials, and each segment is so much important. For example, you know, the prices of urea are high, the prices of DAP, you must be getting the news of these prices. So the alternative is biofertilizers, and in Pakistan, still 1% of the market is not for the biofertilizer. So if we can have, like in the next 5 years, 10%, if we capture that market, how much job opportunities we can create for our young people. So there is a lot of opportunities for Pakistan, and HEC is supporting, Pakistan Science Foundation is supporting, government is supporting to our graduates that they should submit their ideas, they can go for NAC, they can go for like their ORIC in the university. So there are a lot more opportunities are coming, and you should like go for all those opportunities if you are interested to start your own startup.
Thank you again, Dr. Aftab. And about the session that you are mentioning, we will surely like to have you over sometime later. So very quickly, Dr. Saeed, the same question to you. What are the opportunities you see for life sciences graduates?
I think in the current pandemic, many of the opportunities for many of the fields decline, and they are on furthermore. But fortunately for life sciences, we have the opportunity become increased. We have a lot of opportunities, particularly in the diagnostic area field. We have a lot of investment in the laboratory from the government as a private. There are hundreds of labs open now, and they have installed equipment, and they were screening a different level for the patients, for screening at airport or travelers. So the number of opportunities for the life sciences, particularly who linked with the lab, clinical lab, increased. And there are new opportunities, like we never thought about that life sciences people can come in that area, even in the media. You see, there are health programs, they invite the professional, and the anchor, usually they have knowledge about the health or the science. So the new area that been identified that never we seen like doctors or physician usually off and on come on the TV, But these days, even people who have knowledge about the bacteria, virus, or the COVID, they can go and come on the media. So opportunities are there, but look for the opportunity. Like, you need to create the opportunities. If you are just looking for opportunity lying over there, they will be very limited. But if you are smart enough to create opportunity for yourself, then there are too many. You can, but I will emphasize that students should complete their education first. If you are, and you should decide first what you have to do, because if you want to do PhD or MPhil PhD program, so you should try to do it as soon as possible. The earlier the better. And then you are joining a field, then you should decide also that you are joining the pharma industry or any of the diagnostic lab. So you should think about, and there are many opportunities in Pakistan. Luckily, we have quite few diagnostic labs, research opportunities, now the grants. And this field of the diagnostic, not only in the clinical lab, but the equipments, people there import, export the equipments. Over there you have application specialists. So the life science graduates are very much very good paid position over there. So these are another area that been increased during this pandemic. So quite huge challenge as well, but opportunities are there, and smart graduates could find out those opportunities for themselves. So you need to be prepared and smart enough to find and look for those opportunities.
All right, thank you all three of you for sharing your thoughts and answering the questions. So we are opening the forum for the question answers. We have a short time for this because we have to again commence the technical session. So we'll be taking questions from the audience. Please do mention whom you are addressing to in terms of panelists that you want to ask. Ji, Sahida, go ahead.
Assalamu alaikum. My name is Saida and I am BS Biotech student. My question is from, I guess, from Sir Aftab, who is sitting in center. So my so basically मेरा question ये है कि जो जैसे कि हम graduate हो रहे हैं, तो हमारे mind में एक ही question होता है कि यार अगर मुझे आगे कोई scholarship के लिए apply करनी है, so should I go for a job experience or continue my studies? तो मैं different approaches ले रही थी, तो still I'm confused कि अगर मैं आगे continue करना चाह रही हूँ तो मुझे job की requirement है या नहीं, और मैंने ऐसे requirements देखे जिसमें as a graduate को 1 to 2 years की experience required होती है. तो ये चीज़ थोड़ा सा अगर आप clear कर दें तो thank you sir.
Ji, thank you for asking the question. This is actually the question from most of the graduates. They do ask that whether we should go for some job after the graduation or we should continue for the higher education and scholarship. So definitely, if you don't need a job after, like if you don't have to immediately support your family, for example, then you should go, as Dr. Said mentioned, that you should go for your higher studies immediately after your graduation. But you should prepare yourself that you can win a scholarship, and for that, like, it's very important. You should have a publication. You should have done your English language test, for example. That's a requirement in most of the countries. You have some internship experience. That actually matters a lot. So if you have some internship, that also help you. Like, we have a professor from UCL. He can also explain to you that what they look for, like how you can prepare your application in a way that they accept you. But this is my personal experience, being like after interaction with so many experts in different countries. I do ask them the question, but they say that if somebody has some good publication as a first author, they do prefer that person, and they also look for like other skills, like if you are if you can mix very easily with that culture, if you can work in a team, if you have some like experience, if you have done some internship, that actually are the added points that you can build up in your CV. But the important thing is you should have good grades on one side, could have a good publication, should have done the English language test, then you can win scholarship in most of the countries. And you should go for that. And even in that case, you can also support your family, because if you can get a scholarship from like UK, or if you can get a scholarship from the European countries, they pay you enough money that you can support yourself there, and you can also support your family.
right, any other questions? Yes, one at the back.
Assalamu alaikum. My name is Dhuru. I have a question that in which semester or after which semester should we go for internship? Like in final year or in third year, which will be the best for us?
I think somewhere in the middle or just before your last semester is good enough that you can connect your education versus the market. So just before the last semester would be the ideal, because sometime you just end up with your education and going again for internship, then you cannot focus within your studies. So if you the third semester if you have fourth semester, then the third semester would be the ideal, that you come back and again restudy your syllabi and look for the knowledge that is required in the market if you are going to diagnostic lab, research lab. And it will also provide you the opportunity of research, because when you come back you can rethink that research is usually not that difficult. Sometimes we look for hi-fi research, but you should do what you can do. So within the third year you can talk to many people, like back if you have done your internship, over there you can find a mentor who can guide you that how to conduct a research. So if you have done internship plus a little bit publication in your contribution in research, that will allow you, when you're done from your degree, then you are ready for like any of the job, or you will have extra qualification experience with you, and you will be ready for to appear in the interview. So that I will recommend just before the last semester with the good time.
Dr. Aftab, would you like to add some?
Yes, I scanned actually Professor Saeed. He mentioned quite rightly that I personally think that if you have to go for research, for example, then you should go for it early. Like if you are in third or fourth semester, you should have some like background knowledge about the subject, and then you can contribute better in that, like whatever the field you are going to, if you are going some in like food industry, or if you go in diagnostic center, if you will have background knowledge, that will be added point. So for that, like if you can get the time in like in 4 to 6 semester, that will be beneficial. And then if you have your own research project, then you can even tailor according to like what you have learned in there and what are the gaps in there. And with that, you can work on your like subject, whatever you are going to do. Even you can, if you are very good in that, you can also have your own startup. So that could be an added point. So this is how you can also take another journey, like rather going into the job, you can actually find out the gaps in that sector, and you can build something from that. Another point of I would like to add from the internship, I know so many people who have done the internship in some company, especially in the private sector. They were hired. So I know one of the person who actually was an internee in one of the organization in Islamabad. So he joined as an internee, and right now he's the director of that institute. So this is how you can actually build up. So you learn the things, what's going on there, you add value in that organization, and they promote you, and then you can move to a higher segment.
I'm Muzammil, and my question is Sir Saeed. So my question is that how much of a disconnection do you think there's between the industries and the universities? Because we are unable to find enough workshops or trainings, and even if they're in the university or outside the university, to develop our skills and to prepare for the industry, and most importantly to build a network.
Very important question, and this is very like a right scenario, that we have a big disconnect between the industry and our academia. And you are right, the students are not responsible for it. This is the lack of the institution and bridging between the two. Whatever research usually we conduct, this is my emphasis, that the academia must make sure that whatever project are based, it should be on the requirement of the industry. There should be crosstalk between the academia and the industry. Usually there is not. We usually have independent research projects, or topic or syllabus is entirely independent. Whatever HEC usually asks us, we have just sit down and have the outline, and there is no involvement from the industry. So few of the institutions, they have started this, that they focus on the, during the syllabus, they also focusing some of the topics that should link with the industry. And the research base, like in our university, Dr. Mushtaq is here, many of the biotechnology graduates, they are doing a lot of research which is now connected with the industry. So this is the responsibility of the institution and the mentor, that they should connect the research or the study or the academics with the industry. And that also support the student, because if you can able to resolve some of the issue of the industry, like of the pharma or any of the biotechnology product or the veterinary industry, if you've been able to produce some of the enzyme or to resolve their issue, it will also financially help the institution as well as the students. So we should encourage the culture that there should be, like in this, we have the person from the pharma industry. So in such conferences, we should invite many other industry representatives, so we can talk to each other and then should decide for the benefits of the student. This been already started, but we need to increase this, the concept of the biotechnology hubs and other different terminologies are now being biotechnology basket, or different terminology that the industry and the academia sit together and they make a project for the benefit of both.
All right, so we are going to take one last question.
Sir, I have a question from the one who belongs to the pharmaceutical industry. So basically, as a life sciences student, I usually see that business students को ज़्यादा opportunities मिलती हैं in pharmaceutical industries and some sort of other industries जहाँ पे भी R&D की requirement होती है. So my question is, जैसा आपने mention किया कि microbiologist और बाकी जो भी आपने दो other fields को mention किया, तो हमारे as a biotech graduate हमारे लिए वहाँ पे कौन सी opportunity होती है?
बेसिकली फार्मा इंडस्ट्रीज़ के अंदर साइंस ग्रेजुएट मैंने बताया कि फार्मासिस्ट, केमिस्ट और माइक्रोबायोलॉजिस्ट टेक्निकल फील्ड से रिलेटेड हैं. उसके अलावा अकाउंट्स हैं, मैनेजमेंट हैं, एमबीए वाले स्टूडेंट्स भी रिकमेंड करते हैं एचआर में, मैनेजमेंट में. बाकी ये तीन हैं, फार्मासिस्ट, केमिस्ट और माइक्रोबायोलॉजिस्ट. इनकी अपॉर्चुनिटीज़ फार्मा के अंदर बहुत अच्छी है, बहुत वाइटल फील्ड है.
All right, so I said one last question, but I have to take U-turn. So one last question, since one of the speakers want to ask the question.
Assalamu alaikum. My name is Abdullah.
Abdullah, you can ask later. Okay. Dr. Mumtaz, one last question from you.
I'm Dr. Mumtaz here. Dr. Hamad asked a question from Jubair and Sir Aftab. Our graduate from life sciences, where he has or she has to apply. Jubair Sahab said there is only opportunities for microbiologist, chemist as well as pharmacist. But there is no lack of or short of opportunities for life science students. And there's Sir Aftab said there's a problem for a graduate from the life sciences. He or she has to apply for a scholarship as well. As we know, for a scholarship, there is not in a huge quantity or not a huge amount scholarship for everyone. First author paper problem here. If a graduate, fresh graduate has no experience, as well as even though you are familiar from the university's condition, there is a big problem of basic facilities. Even though our graduates not able to know the titration as well as, even though sir from the UCL, there is a problem of prescription of the doctors in hair as well as waste. Even though we know there is biotechnology as an environmental science as a vast field and vast application in the different areas in the different countries, but we are unluckily getting some focus on this field. Okay. I think I am not satisfied from this question answer as well.
Well, it's a more a comment that Dr. Saab, he has mentioned that actually there are limited opportunities for our young researchers, especially who are working in our universities, and the condition is not that good. But I personally feel that if you want to go for a scholarship, you can opt it, and I know that there are, like even in China, there are around 10,000 Pakistani students, those who are studying there. So there is a lot of opportunity. China is offering a lot of scholarship. Korea is offering. Malaysia, you can do PhD in 3 years. So they are offering a lot of scholarships. So even if we focus on these countries, there are a lot of opportunities there. The other thing I said that you should actually identify the problems, and you can also go for your own startups, and there are a lot more opportunities now. So you should like contact the student. They could be in touch with the ORIX, and they even with the NIC. So from those things, they can also have like more opportunities. But you have to prepare yourself, as Dr. Saeed mentioned, that you should build yourself in a way that initially you should decide what you are going to do, and then you should build yourself up accordingly. And there are a lot of opportunities. If you are like good enough, everybody will be looking for you. And in Pakistan, industry person is there, so he can understand that. I know there is a survey from Pakistan. They were not satisfied by the performance of our graduates. More than 90%, with more than 90% like the graduates, they were not satisfied. So we are not preparing them enough. So the important thing is from the students' ends as well as on the faculty end. So they both should make effort that they should be up to the standard. And if they are up to the standard, opportunity will follow them. They don't have to follow the opportunity.
I think Dr. Mumtaz is not yet satisfied with the answer. He will give his own answer during tea break.
All right, so on that lighter note, we would like to conclude. So on behalf of organizing committee of the conference, I would like to thank Professor Dr. Saeed Khan, Dr. Aftab Ahmed, and Mr. Jubair Ali Watio for your presence, and thank you for joining us. So this concludes our panel discussion session. Thank you so much for your presence. Thank you.
Hello. Hello. वहाँ से खोलो गेन इसको, उधर से गेन खोलो. डॉक्टर आसिफ कुरैशी. आवाज़ आ रही है? हेलो आ रही है ना उधर? हेलो. आप सही हो? हेलो. या लगा गया, तुम एक्सपर्ट हो ना? कोल्डस में किसको लगाना? कोल्डस में काफी सारे जो भी स्पीकर आए उनसे पूछने प्रेसिडेंट के लिए नेट का मसला नहीं, वो तो सुबह था. छोड़ तो तनवीर देख लेगा बाकी वहाँ पे तुम इधर अबे बंद तो करो बंद तो करो
Hello, hello, hello. A very big thank you to all our esteemed A very big thank you to our esteemed panelists for gracing us with their presence today. To show our appreciation, I would like to request Professor Dr. Kashif Ali, Dean Faculty Life Sciences, onto the stage to present the panelists with shields. Panelists, please make your way to the stage as you hear your name. Professor Dr. Saeed Khan. Mr. Jubair Ali Watio. And Professor Dr. Aftab Ahmed. Once again, thank you to all our panelists. Moving forward towards our plenary lectures, I would like to call to introduce Professor Dr. Mushtaq Hussain. Professor Dr. Mushtaq Hussain has done his PhD in genetics, genomics and systems medicine from University of Glasgow, UK. Currently, he is the principal at Dow College of Biotechnology, Dow University of Health Sciences. Sir, please make your way towards the stage for your presentation. A round of applause.
Thank you very much for providing me this opportunity, and thanks for the organizer and the introduction. So I'll be speaking about COVID-19 host and virus genetic variations. Now, possibly you might have seen this photograph. This guy, his name is Charles Darwin. In 1859, he famously published a book named as Origin of Species by Means of Natural Selection. What that book actually explained the very basic enigma of the life sciences from the very beginning. Why there is such a diversity of the life, how this diversity at the first place have been originated, and how it then transferred or has been disseminated to the different demographies or different geographical areas of the world. That's the book. And then in 1944, he developed that particular tree, which shows that previously the version was that the life originated from the least advanced to the most advanced level. Gradually, there is a ladder going from the less complexity to the more complexity. What Darwin actually presented the idea, this is not the case. Actually, life diverged in variety of different ways, and some of those variations have been taken advantage from the, have actually been selected out from the environment because of some particular advantage. It may not always be the best of these species that comes out to be the most sustainable one. It's just one particular trait that defines the survival or the demise of a particular species. And he put a very powerful word over it, and he says, I think. That actually changed the way we see the life sciences as of today. As the Richard Dawkins actually quoted it, evolution is the greatest show on earth. And one of the picture or one of the film, or you can say one of the evidence of this greatest show on earth we have seen in the last 2.5 years. We called it COVID-19. COVID-19 has been caused, as you all know, because of the virus which is called as SARS-CoV-2. It belongs to the beta coronavirus, RNA containing virus. These are the few strains of the beta coronavirus that are known to infect human. There are 7 of them. Only 3 of them have certain particular property that it provides them a selective advantage to cause serious disease burden or serious health concern. First is the SARS-CoV that has caused the epidemic back in the year 2003, MERS-CoV from 2008 to 2012, and quite recently SARS-CoV-2. They all are being originated from a single common ancestor. Slight genetic variation provided one advantage to the other, slight one advantage to one particular strain of the virus, to other, and now we know SARS-CoV-2 have several strains of the viruses because of such genetic variation. And what I'll be talking about are those variations and the variations on which we have worked at the Dow University of Health Sciences. So origin, as far as the origin is concerned, now since I have a very short amount of the time, so I'm going to skip the origin of the virus. In summary, the virus is not being man-made, it's not a laboratory accident, it's being naturally evolved. We know that because there are some signature signs within the spike protein of the virus that belongs to the human coronaviruses as well as the bat coronaviruses. If somebody have to mischievously design the virus, this is one of the very bad way of designing the virus for any mischief activity. Next slide. Yeah. So we started our work from this paper actually. That paper has been published in April 2020, and what it shows that how SARS-CoV-2 virus, a protein of the SARS-CoV-2 virus, at the first place binds with the human protein. So the gold which you are seeing over here is a SARS-CoV, part of the spike protein of the SARS-CoV-2. The purple which you are seeing over here is actually the human ACE2 protein. That's where the interaction is happening. Now we know that this particular motif that is called as RNA receptor binding motif, that is actually being involved in the interaction of the spike protein of the SARS-CoV-2 with the human ACE2 protein. So now this is actually the region of that SARS-CoV-2 spike protein that binds with the human protein. That's the S2, which has a peptidase M2 domain. So we confined our research to only this particular region. Since the virus has been evolved in humans, possibly in those peptidase M2 domain, in different humans there are slight variations may be present that may impair the binding of the virus at the first place. So then about 200,000 genome projects database has been analyzed, and what we have found that these are the few interaction points where the virus spike protein binds with the human ACE2 protein. And humans have variations present in different ethnic groups. So then we explore whether those variations confer some degree of the resistance against the attachment of the virus or not. And if these variations are actually leading to the impairment in the attachment of the virus, that may confer the natural resistance against the SARS-CoV-2 infection. So these are those variations. Now you can see These are being listed over here. S19P means serine is being present as a normal wild type among the human population, but certain individuals have, instead of serine, there is a protein being present there. So that provides, and this serine is actually the attachment site for the SARS-CoV-2. So those individuals who have a serine being present at that 19 position within the S2 protein, the SARS-CoV-2 is binding very fine. But if it is being mutated into some other residue, so in that case, what's going to happen? The virus is not going to bind. So that's what we have explored. So the first thing what we have seen is actually modeled all of those variant proteins, and then we superimpose over all of those variant protein over each other, and what we have found that their backbone is more or less very much similar. However, there are some spatial variation which we have observed compared to the mutated residue and the original one which is present in the wild type. So the green what you are seeing over here are the wild type residues. Is there a pointer here? No pointer. The green which you are seeing over here is a wild type residue, and the purple which you are seeing, or the blue, if it is appearing as a blue, blue is actually the mutated residues. So there are certain residues, say for example here you can get the spatial variation there, and many other residues, they are being more or less superimposed. So then we explored the binding. So this is the spike protein of the SARS-CoV-2, the part of the spike protein receptor binding region of the SARS-CoV-2 spike protein, and what this is actually the variant proteins. And what we have found, there are variations in their intermolecular binding, and that is the variation lies in the polar-polar, apolar, charge-apolar interactions. And then we further analyze on the basis of the thermodynamical values. And what we have found is actually three residues, three type of the mutations, that is the S19P, and mostly actually two, that is S19P and E329G. These are the E329G. That actually shows less efficient binding with the SARS-CoV-2 spike protein. So it means individuals who have those mutations are naturally resistant against the binding of the SARS-CoV-2. This is not the first case where the humans naturally have been found resistant to the SARS-CoV-2 infection or any viral infection, naturally, without the support of the immune system. It's actually the genetic background defining the resistance actually. One of those cases, very famous one, is actually some of the prostitutes in Africa are naturally resistant to the HIV. Why? Because the receptor which is required by the HIV to bind, they have a mutated receptor there. So it doesn't matter how many times they have been contacted with the HIV, simply the virus can't attach to their cell. So it's not the immune-driven resistance, rather than the genetic background defining the resistance against the virus. So that possibly be the reason. And we know that these alleles are being present in different proportion in different populations. That might be the explanatory factor that maybe in the Pakistan, despite much less of the safety issues have been taken, still the virus have not caused that much havoc as we were anticipating earlier. And that work has already been published in Journal of Medical Virology in 2020, and it's already being cited. Patients actually may confer the resistance against the attachment of the virus, then the virus penetrates into the cell. And for the penetration of the virus, what happens, one of the human serine protease that is called as TMPRSS2 actually cleaves the spike protein. That's what we call priming. And once it cleaves the spike protein, it allows the virus to enter into the cell. Since TMPRSS2 is also the host protein, variation in the TMPRSS2 may make them unable to prime the viral spike protein. Thereby, virus might be able to attach with the human cell, but may not be able to penetrate inside it. That again confers the natural resistance. So first we explore, this is the extile analysis, and it shows that this particular protein has a very low probability of the crystallization So it means, doesn't matter how hard you may try, you may not be able to get the crystallized version of the protein, so empirically you cannot resolve that structure very quickly at least. So then what we have done, we have modeled this particular protein. This is actually the protein of the model of the TMPRSS2, and then we docked it against the spike protein. So there are the two regions which we have identified that this TMPRSS2 of the human actually binds with the spike protein. So first, it provides a possible target to develop a drug against these particular two attachment sites. So say for example, if you develop a small molecule, what's going to happen? The small molecule is going to interfere this interaction. That's how the TMPRSS2 may not be able to cleave or prime the spike protein, and the virus may not be able to enter into the human cell. Second, mutation in those interaction sites, the purple one, may also confer the resistance against the virus. So that's what happened in the Iran, sorry, I have not included that slide. In the Iran and Italy and Spain, there are 3 studies which have been conducted, all prove that some of those interaction sites are, if being varied in the human, may confer the resistance against the invasion of the SARS-CoV-2 within the cell. The next we start looking for other targets. So that paper is a very fantastic paper, and I really recommend you to read this paper. It's seriously good paper. That paper has been published somewhere in April 2020 again. So what they've done, they've actually taken out of the 29, 26 genes, they have cloned it into the individually into the cell line, and then by the co-immunoprecipitation method, they have identified what are the human protein that binds with the 26 different proteins of the SARS-CoV-2. And they end up with this. These are the 417 proteins of the human. They are known to interact with the SARS-CoV-2 proteins, 26 of the human, of the SARS-CoV-2 protein. So this is the combined network once we have constructed the combined molecular network of it. Then we start looking for it, how to model the disease in a safest possible organism. So one of my very keen interest, and in fact one of my prime interests now, is actually a small little humble fruit fly. It's a 2 millimeter size fly that is being effectively known as a disease model or the model for the genetics. You might have heard this fly name as Drosophila, right? So what we have actually explored, that out of those 417 proteins of the human that interacts with the SARS-CoV-2 26 protein, how many of them are actually being present in that small fruit fly? What we have found, out of 470, 380 proteins are direct orthologs with the human proteins. So it means this small fruit fly could be used to model the disease biology of the SARS-CoV-2. And in fact, some of the experiments we have done, which are quite encouraging as well. That data has been published in Data in Brief earlier. The previous one has also been published in Ames Microbiology. Then a variant arises. As I said, evolution happens. It's a continuous process. The virus. There are so many of other variations which are happening at the period of time, during the period of epidemic. At this moment of time, what we know, there are more than 50,000 variants of the SARS-CoV-2. Out of those 50,000 variants, only very few of them got notoriety, if you like. One of those, the first famous case is of the B117. Now we know it as alpha variant of the SARS-CoV-2, or the UK variant. What we, once the genome has been sequenced, what we identified that there are about 26 different mutations which are being spread throughout the 29 genes of the SARS-CoV-2. And some of those variations are being present in the spike protein, and some variations are present in another protein which we call ORF8. Why we got interested in the spike and the ORF8 protein? Because these are the two most immunogenic protein of the SARS-CoV-2. Spike protein, quite frankly speaking, most of the vaccines are currently based on the spike protein. So that's one thing. The second thing is actually the ORF8. There's a paper in the Nature Scientific Reports, and if you see over there, it produces the antibodies which remains in our body for a much longer period of time than the spike protein. So it's a very fine diagnostic tool if you like to use it. So then the first thing we have checked the B117 spike protein structure of the SARS-CoV-2, the alpha variant spike protein structure, and the wild type of the Wuhan strain of the SARS-CoV-2 spike protein. More or less the structure is pretty much same. The residues are slightly differentially oriented. The binding with the ACE2 receptor is not being affected. However, what's been changed is actually the antigenic spot, or we called it epitopes within the spike protein. What we have found, a particular epitope which has been present at this particular region, and why this region is very important, I'll explain you in a short while. Now see here, that's a co-crystallized structure of the surface topology of the SARS-CoV-2 spike protein. This is the spike protein. And these are the human antibodies. The green, the sea green you are seeing are the antibodies which are binding with the spike protein. So you can see that the spike protein, the antibodies of the humans are binding with the two different regions of the spike protein. One which is called as N-terminal domain, the other one is the receptor binding domain. So in the human, we produces the antibodies against the SARS-CoV-2, and these antibodies are binding to the two different regions of the spike protein. One is the N-terminal domain, and the one is the receptor binding domain. The mutation which happens in the alpha variant are actually in the N-terminal domain. So if you can see over here, the gold and the purple is pretty much superimposable to each other. So whatever the mutation which are happening in the receptor binding domain in the alpha variant are not affecting the binding of that particular spike protein with the antibodies. However, if you see over here, the gold and the purple are not very much superimposable, especially in this region and in that region. What it means that the N-terminal domain structure of the alpha variant has been changed compared to the Wuhan strain. So it means it may impair the binding with the antibodies. And that's what we exactly have found. And later on it has turned out to be that some of the individuals who have been previously infected with the SARS-CoV-2 Wuhan strain got reinfection by the alpha variant. One of the classical reasons could be that then the virus is evading the already existing immune response because of the variations there. That could not be the only reason, but that could be one of the reasons. Then the next protein is the ORF8 protein. That's a very interesting protein. Why? Because that's the Wuhan strain spike protein, ORF8. All those colored blob, if you are seeing over here, these are the antigenic region of the ORF8 proteins. Okay? This is the SARS-CoV of the 2003 strain ORF8. Now, the B117 alpha variant has lost considerable number of the epitopes compared to the Wuhan strain. So again, that's an immune evasion mechanism that virus may have evolved during that 1 year of the epidemic in by the September 2020. That allowed them to survive more in the humans, or disseminated more in the population. That work has already also been published in Journal of Medical Virology. Next. Then we started working with our Malaysian collaborators. So what they have done, they have actually sequenced the whole genome. They have sent us the sequences, and some of the sequences which we have done over here, we have also sequenced it by the next generation sequencing. The whole genome of the virus have been sequenced, and what we have found, the 3 different new lineages of the viruses, which have not, of the SARS-CoV-2, which have not been previously identified, and they have actually been now labeled as Malaysian lineage. So that work has also been published in PLOS One. Now what is the current status? The current status, what we know, that there are some variants of the SARS-CoV-2 which are labeled as variants of concerns. And there are some variants which are being labeled as variants of interest. All of those variants have one or other mutations being present in their spike protein and anywhere else in their genome. And these are, and now we know already there are breakthrough cases, there are escape cases, there are vaccines which are not being efficient. Say for example, as far as the current Wuhan vaccine is concerned, whether it's Pfizer, if it's Pfizer or it's being Moderna or it's being Sinovac or Sinopharm, these vaccines have already different degree of the efficiency against the Wuhan strain of the virus. But all have been shown that the efficiency against the variants have been dwindled. Say for example against the alpha variant, the first variant, previously when we have said that it might escape from the vaccine. So there was lots of hue and cry about it, that why I have said that thing. Even Saeed actually met me, what you have said to the media. I say I have said it might. actually escape from the antibodies which are being elicited by the vaccination. And it does. The latter on it has been proved that the Moderna and the Pfizer have 20% less efficiency against the alpha variant. According to some results, about 30 to 35% against the beta and gamma. Against the delta, 25%. Omicron, 23%. So they have much reduced efficiency against all of those variants. And that could be the case that you have been vaccinated but still you got the SARS-CoV-2 infection. It could be a different variant of the virus you are getting. So we thought that could it be possible to design a vaccine that could entertain or that could curtail all of the currently circulating variants of the virus and possibly the future one. So what we have done, we have actually taken the two particular regions of the SARS-CoV-2 spike protein, as I have said, that is of the N-terminal domain and the receptor binding domain to which the antibodies are binding. These are the epitope maps. The red regions, the red blobs are indicating the epitope, so the antigenic region of the SARS-CoV-2 spike protein. These are of the Wuhan, and this is again of the Wuhan. N-terminal domain, receptor binding domain. These are the same region of different variants of the SARS-CoV-2. Now you can see there are subtle variations being present in the N-terminal domain, epitopes, as well as the receptor binding domain. And that's so out of those variations, we thought that could a particular hypothetical sequence of the N-terminal domain and the receptor binding domain could be designed that could have all of those variations in the epitope in a single molecule. And that's what the hypothetical N-terminal domain and the hypothetical receptor binding domain. But once we have identified that, we modeled it, and then we combine them. So that's the hypothetical receptor binding N-terminal domain, that's the hypothetical receptor binding domain, and we link it with the IBNA1 gliala protein, a small portion of the IBNA1 gliala region. That's a repetitive glycine and alanine region which are known to protect the IBNA1 protein, that's from the Epstein-Barr virus, to not being degraded by the proteasome. So once this particular molecule is being synthesized or being actually translated into the human, the protein is not going to be degraded, and it's going to protect it, this particular small peptide of the glial repeat. The N-terminal portion we have to add a tag sequence and for the nuclear localization, because we want that particular mRNA to go inside the cell and at times inside the nucleus as well. So that's the combined structure of the protein. And then we have checked their RNA stability. So if you can look over here, this is the second three-dimensional structure of the Pfizer molecule. This is the three-dimensional structure of our mRNA construct. So briefly speaking, this is the mountain plot. So what we know that to understand this particular graph, there are three lines, red, blue, and green. Red, blue, and green line, the more those lines are being superimposed over each other, the more better stability the RNA molecule has. This is the stability of the Pfizer molecule. This is the stability of our mRNA construct. Next, now that's the immune simulation. So immune simulation, so I will not go into this detail of this graph because I have been told that I have to finish it. Just focus on this yellow line. This yellow line represents the antibody titer. With the Pfizer vaccine, this is the Pfizer vaccine, the antibody titer within the 15 days is about 5,000 molecules. 35 days, it will be dwindled to between 2,000 to 3,000 molecules. The R construct is producing the immune response up to 12,000 molecules in 15 days and up to 30 molecules between 6,000 to 8,000 days. Now that work has already been, we have submitted it, we got the revision of it, we have submitted the revision and it's in press now, and shortly in about a week time or so you may see the paper of it. Now these are the individuals who have contributed a lot during this work. This is my dear colleague Nusrat, Basma, Aisha, Anusha, Sanya, Fauzia, Nasir. Nasir is from IBA, he's from the computational side, and this is the biochemist, microbiologist, biotechnologist, all three of them. And at that point of time, these three were undergrad when we have done this work. Thank you very much. Shall I?
Sir, is it possible that we can prepare restriction binding domain? जैसे आपने कहा था कि enzyme restriction binding domain पर वायरस एक HIV पेशेंट था, वो रेजिस्टेंस थी उसके अंदर restriction binding domain पे, जिसकी वजह से वहाँ वायरस उसके बाइंड नहीं हुआ, तो जिससे HIV रेजिस्टेंस थी उसके अंदर. कैसा पॉसिबल है कि जो डिफरेंट वायरसेस हैं, ठीक है, तो उनकी भी रिस्ट्रिक्शन बाइंडिंग डोमेन होगी बॉडी के अंदर, तो हम कोई ऐसी ड्रग प्रिपेयर करें या उसके अगेन एंटीजन के वो रिस्ट्रिक्शन बाइंडिंग डोमेन को वो वायरस टारगेट, वो पहले से हम वो ड्रग टारगेट करें, जिससे आने वाले जो वायरस हो उस बाइंडिंग डोमेन पे बाइंड ही ना हो, जिससे वायरस का इन्फेक्शन ही स्प्रेड ना हो.
Alright. So what is actually a receptor binding domain?
Receptor binding domain.
Okay. So yeah, hypothetically speaking and theoretically seems nice, but let me ask you one question very simply. So the virus is exploiting one of your protein and a part of that protein is a receptor binding domain, right?
Yes.
For its advantage. Why that receptor binding domain and why that protein is present in your body in the first place? Because of certain particular function.
Yes.
If you design a drug for no use, because at this moment of time you are not being infected with the virus,
yes,
and you're just taking this drug, that might affect the normal physiological function of that particular protein. So theoretically, yes, you can design a molecule against binding with any particular molecule. There are about 23,000 proteins being encoded by your genome. You can design any small molecular inhibitor against any of those 23,000 molecules. But is it being necessary? Because those 23,000 proteins are because of some particular evolutionary advantage. Virus is using that particular advantage of yours in its own advantage. That's how it works.
तो जो HIV पेशेंट था जिसके अंदर वो रिसेप्टर absent,
deleted. Part of that particular receptor has been deleted. CXC4.
So that's why HIV virus is not binded. So उसके अंदर कोई side effect नहीं हुए थे?
Obviously हुए होंगे. That I am not aware of. But let me tell you, this is what we call in evolutionary biology trade-off. You got an advantage on one side, but because of that particular trait, you got a disadvantage as well. Say for example, you got a larger larger cranial cavities. Okay, why? It provides you a bigger very big advantage. What is that advantage? You can accommodate a larger cerebral hemisphere that defines your intelligence and cognition and many other things, right? That is an advantage you got. But at the same time, humans, female, require assistance in delivering a baby. You got a disadvantage. Human mortality without medical intervention, if you exclude all the medical intervention in the last 200, 300 years, human mortality during the childbirth is far higher than any of the other mammals, including primates. Why? Because one of the reasons is actually the larger cerebral side. So you got an advantage on one side, but on the other side you got a disadvantage. So evolution is a complete blind eye, and then the nature will select what is being fruitful to you. So CXC4, consider a possibility. Would you like to have a diabetes or be died? Died. Well, you have a very morbid desire to face the end of the world, isn't it? Diabetes is much less threatening than being dead, right? So consider a possibility that this HIV virus get airborne. It is spread like SARS-CoV-2 or any other of the flu virus. What's going to be happening then? That in about 5-year time, all of those individuals who actually don't have that mutations will be dead because of the HIV infection. Pharmaceutical companies cannot cope with that much amount of the drugs against the HIV, neither that much mass vaccination could be taken. Okay? So in 5 years' time, a single mutation in the virus that possibly could make HIV airborne could change the gene pool of a much advanced, or possibly, if you like to say, the most advanced species on the earth. Previously, those particular CXC4 mutations are considered as a mutated individuals. But once that 5-year period has done, now all of those wild type are dead because of the HIV, and now the CXC4, which was once the mutated one, becomes the wild type. So that's the force of nature, or evolution if you like.
Thank you, sir.
You're welcome. Shala.
Thank you, Dr. Mushtaq. I would like to make a small announcement for the rest of the speakers. Since we are unfortunately short on time, so all of the remaining speakers are requested to please abide by the time frame, which is 15 minutes for each presentation. Moving on, our next speaker today is Professor Shah. The next lecture is going to be delivered by Professor Dr. Raza Shah. He is working on the area of nanotechnology and supramolecular chemistry. He was declared top scientist for the year 2019 by Directorate of Science and Technology, Government of KPK. He received the Civil Award Tamgha Imtiaz 2015, Atar Rahman Gold Medal 2006, and Dr. Raziuddin Gold Medal 2015 by Pakistan Academy of Sciences. He received Abdus Salam Nobel Laureate Award and selected TWAS Young Affiliate in 2010 by Third World Academy of Sciences. He has authored 5 books and also edited 2 books, along with 7 chapters in books and more than 430 research articles in international journals with impact factor of more than 1,330. He has 2 US patents to his credits as well. He has supervised and co-supervised 33 PhDs and 43 MS MPhil scholars. The research projects of Dr. Shah are funded by both national and international funding agencies. Sir, I would request you to please make your way toward the stage to deliver your presentation. Thank you.
Okay, A'udhu Billahi Minash Shaitanir Rajim, Bismillahir Rahmanir Rahim. First of all, let me thank the organizer and my student, Khairbakhsh. He is not, I can't see him, but he invited me, and Mumtaz is also here, and Jubair is again here. So thank you so much for inviting me to just share some of the information about the work which we have done, and also about the thrilling world of discovery. So in next 10 to 15 minutes, I would be talking about the thrilling world of discoveries. As Dr. Mushtaq talked about more Wuhan virus and beta and these viruses. So these are all actually nanomaterials, or I would say these are the materials which you can term as nano. So the virus is also nano. So that's why this world, there was a time when these microchips were developed. So it just revolutionized the whole material sciences. But now a new paradigm shift is observed after the nanotechnology. So that what in this world, we are living in such a world where there are poor peoples and there are peoples who are having plenty. So that's what is the reality. But how we can equalize these both? There is only one way to do that, and that is that we should invest on science and technology and discovery. So science and technology is the only way through which you can just reduce the gap between the poor and the richer. So that is why that our government and especially government from the Islamic world should invest more on science and technology. And as you know that the socio-economic development is no more dependent on the natural resources. So there was a time when we were just, our elder were telling and our older teachers were telling in the school that Pakistan is an agriculture country and we should just invest on agriculture. But it's no more the reality that if you are investing on agriculture and natural resources, you would not be included in the developed world. You would have to invest on the science and technology, and that's why I would be just giving you some glimpses and then a little bit about our own research work which we have conducted in our center. Somehow this is not working. Is that we are living in the world that truth is stranger than fictions. That's what I used to tell my student. That there was a time when the people were telling us that one can disappear. Harry Potter cloth. आप लोगों ने हैरी पॉटर के क्लॉथ को तो वो जब वो चोगा उड़ देती थी ना तो डिसअपियर हो जाती. तो ये एक फैक्शन था, लेकिन अभी जो है जो मेटा मटीरियल से जो कपड़े हैं वो अगर आप पहन ले तो आप फिजिकली डिसअपियर हो जाएंगे. तो अब ये रियलिटी है कि ऐसे मटीरियल बन गए हैं कि जैसे आप डिसअपियर हो सकते हैं, आप लोगों को नज़र नहीं आ सकते. और ये तमाम जो कमालात है वह दीज आर ऑल अबाउट नैनो टेक्नोलॉजी. नैनो टेक्नोलॉजी ने इस तमाम चीज़ों को यानी एक नए डायमेंशन तक लेके गए हैं. जस्ट एक सिंपल डेफिनेशन कि व्हाट इज नैनो टेक्नोलॉजी. इफ समवन इज वर्किंग or manipulating any material or particles, and one dimension of that material or particle is less than 100 nanometers, so then we say that this particular person is working in the area of the nanotechnology. So this is just for the student, that's nanotechnology. Let me switch to why nanotechnology is so important. Nanotechnology इतनी importance उसकी क्यों है? नैनोटेक्नोलॉजी के इम्पोर्टेंस के दो तीन वजूहात हैं. नंबर फर्स्ट कि ये जो है आपके जो मटीरियल्स है उसके प्रॉपर्टीज़ को चेंज करती है. जब आप माइक्रो से नैनो लेवल तक आते हैं तो मटीरियल प्रॉपर्टीज़ कंप्लीटली चेंज हो जाती हैं. सिंपल एग्जांपल ये है कि आपने अगर गोल्ड के अंगूठी हाथ में पहनी है, आप पानी में डाल दे कुछ नहीं होता, लेकिन यही अंगूठी अगर आप गोल्ड, यही गोल्ड है, यू हैव नॉट चेंज द मटीरियल, यही गोल्ड है लेकिन आपने गोल्ड नैनो पार्टिकल से बना लिया, उसको कंप्रेस किया, फिर पानी में डाल दिया तो फिर आप ढूंढेंगे नहीं मिलेंगे, वो सोल्यूशन में कन्वर्ट हो जाती है. क्योंकि गोल्ड जो है एट द नैनो साइज पानी में सॉल्युबल है. इसी तरीके से गोल्ड जो है इनर्ट है. बाय नेचर जो केमिस्ट्री जिसको थोड़ा सा बैकग्राउंड है उसको है कि गोल्ड जो है इनर्ट होते हैं, लेकिन जब अगर आप इसको नैनो स्केल में कन्वर्ट कर लेते हैं तो वो सुपर कंडक्टर हो जाते हैं. क्यों होते हैं? क्योंकि नैनो में जो बैंड गैप होता है, जब आप जितना भी साइज आप रिड्यूस करते हैं तो क्या होता है कि जो न्यूक्लियस के आउटर इलेक्ट्रॉन पे इम्पैक्ट जो है कम होता जाता है और जो नेबरिंग न्यूक्लियस ऑफ द एटम्स है उनका इम्पैक्ट बढ़ता जाता है, जिससे क्या होता है एक इलेक्ट्रॉनिक सी क्रिएट हो जाती है और उसके बेस पे जो मटेरियल प्रॉपर्टीज है वो टोटली चेंज हो जाती है अगर आप किसी भी मटेरियल को नैनो स्केल में कन्वर्ट कर ले तो. और वजह क्या है क्योंकि सरफेस एरिया आपने बढ़ा दिया। जब भी आप सरफेस एरिया किसी भी मटेरियल के बढ़ा देते हैं तो एट द सेम टाइम व्हाट यू डू, यू इंक्रीज द रिएक्टिविटी ऑफ दैट पर्टिकुलर पार्टिकल। सो सरफेस टू वॉल्यूम रेशो बढ़ जाते हैं और आप जितने भी कैटालिसिस है, जो लोग कैटलिस्ट पे काम करते हैं या जो लोग इंडस्ट्रीज में काम करते हैं, तो वो अपनी जो मटेरियल है या कैटलिस्ट है उसको फाइन पाउडर में कन्वर्ट करते हैं ताकि उसकी रिएक्टिविटी बढ़ जाए। तो ये जो है एक्चुअली अगर आप नैनो स्केल पे लेके आ जाए तो फिर जो है प्रॉपर्टीज कब एब्रप्टली चेंज हो जाती हैं। एंड दैट्स व्हाट इज कि आप इलेक्ट्रिकल प्रॉपर्टीज चेंज कर सकते हैं, मैकेनिकल प्रॉपर्टीज, ऑप्टिकल प्रॉपर्टीज, केमिकल प्रॉपर्टीज एंड फिजिकल प्रॉपर्टीज। जब आप मटेरियल माइक्रो लेवल से नैनो लेवल तक लेके आ जाते हैं तो क्या हो जाते हैं, ये तमाम प्रॉपर्टीज चेंज हो जाती हैं। और यही जो है डेली लाइफ में यूज होते हैं। मसलन आप नैनो पार्टिकल इस वक्त अवेलेबल है मार्केट में, इसको सोल में जो सॉक्स है, सिल्वर नैनो पार्टिकल एम्बेडेड सॉक्स प्रिपेयर्ड हुए हैं जो कि जिन लोगों के स्मेली फीट होते हैं वो अगर ये सॉक्स इस्तेमाल करते हैं तो उनके पाँव से या शू से बदबू नहीं आती। तो ये जो है क्योंकि ये बैक्टीरिया को किल कर लेते हैं। नॉर्मल केसेस में अगर आप सिल्वर यूज करते हैं तो इट्स नॉट फंक्शनिंग लाइक दैट। सो दैट्स व्हाई के नैनो रीजियन में कंप्लीट प्रॉपर्टीज चेंज हो जाती हैं। इसी तरीके से जो ड्रेसेस हैं वो भी ऑलरेडी आर्क ने डेवलप किए हैं इसी तरह के ड्रेसेस के। अगर आप नैनो मटेरियल इसमें यूज़ करते हैं तो इट वुड नॉट स्मेल। और जो जर्म्स है ख़ासकर आज के जो पेंडेमिक के दौर में अगर मास्क और इनमें नैनो मटेरियल बहुत सारे यूज़ हुए हैं, बल्कि अभी जो इस वक्त जो पॉइंट ऑफ केयर डिटेक्शन डिवाइस थी ये कोरोना एंटीबॉडीज़ के लिए, वो भी गोल्ड नैनो पार्टिकल की थी। सबसे पहले जो FDA ने अप्रूव की थी वो गोल्ड नैनो पार्टिकल के जो SARS-CoV-2 के लिए के एंटीबॉडीज डिटेक्शन के लिए होते हैं। और जस्ट ये है कि आप ब्लड उस पर लोड कर ले एंड इट जस्ट सेंस दैट वन। इसी तरीके से जो वायरसेस हैं इनको आप जो है डिजीज पर को ट्रीटमेंट के लिए यूज कर सकते हैं एंड दैट इज आल्सो विद द हेल्प ऑफ द नैनो टेक्नोलॉजी। इसी तरीके से एजिंग को रिवर्स किया जा सकता है। एंड अगेंस्ट जो डेविड सेंक्लेयर हैं उन्होंने यही एक्सपेरिमेंट्स कंडक्ट किए थे माइसिस पे NADH को यूज करते तो एंड दैट्स आल्सो विद द हेल्प ऑफ द नैनो टेक्नोलॉजी। तो जो एज की प्रोसेस है दैट व्हाट वन कैन रिवर्स विद द हेल्प ऑफ द नैनो टेक्नोलॉजी। आई वुड जस्ट इसी तरीके से जो एक कंसेप्ट था एंड अगेन दैट वर आल्सो द साइंटिस्ट यूज द प्रिंसिपल ऑफ द नैनो टेक्नोलॉजी कि ऑर्किड्स के प्लांट्स में लुमिनेसेंट लेके आ जाए और आप जो है ये लाइट देना शुरू कर दे रोशनी में, जैसे जुगनू के जो जीन्स है दे जस्ट क्रॉस विद दिस प्लांट। सो ये जो ultra long release drug delivery system, this is the area where currently I am mostly working, कि कुछ ऐसे पॉलीमरिक मटेरियल है, कुछ ड्रग्स ऐसे होते हैं कि आपको सुबह शाम लेनी पड़ती है या हर दिन लेनी पड़ती है। तो क्यों नहीं ऐसा सिस्टम डेवलप किया जाए कि आप हफ्ते में सिर्फ एक टैबलेट ले लें, काम उसी तरह कर लें जिस तरह की आप रोज़ाना लेके मेडिसिन्स लेते या करते। तो इसी तरीके से नैनो मटेरियल्स के लिए पॉलीमरिक नैनो मटेरियल यूज हुए हैं जिसमें से 15 दिन में सिंगल डोज लेके और वो जो बहुत सारे उसमें चेंजेस हो गए, बहुत सारे ड्रग उसने रिप्लेस कर दिए हैं कि जो लोग हर रोज लेते थे अब वो हर 15 दिन बाद एक टैबलेट लेते हैं एंड इट वर्कड एंड इट स्टे इन योर बॉडी फॉर 15 days। सो दैट इज समथिंग which rationalize। what we have done in my research group, this is just glimpses. हमने तीन बेसिक एरिया में नैनो मेडिसिन में तीन एरिया में काम किया था. एक नैनो मेडिसिन, नैनो डायग्नोस्टिक एंड ड्रग डिलीवरी सिस्टम. आई विल जस्ट डिस्कस वन बाय वन these. This is a bit old result कि इसमें हमने ये किया था कि मेरे कोलीग थे डॉक्टर नूरुल कबीर बांग्लादेश से, उसके साथ मिलकर हमने किया था कि हमने जो रेट्स है उसमें सिरोसिस इंड्यूस किए थे, और फिर जो FDA अप्रूव्ड ड्रग है फॉर द लिवर सिरोसिस सेलेमाइन उसको लिया था, और उनसे हमने गोल्ड नैनो पार्टिकल बनाए थे. एंड देन वी सब्जेक्टेड दीज वाइसेस जो ट्रीटमेंट पे लगा दिए थे. एंड आई वुड जस्ट skip this one, the details, but I just discuss the result. ये जो है जो नैनो पार्टिकल्स जो हमने बनाए थे, ये जस्ट उनके कैरेक्टराइजेशंस है कि वी कैरेक्टराइज द सिलिवायरिन कोटेड गोल्ड नैनो पार्टिकल्स एंड विच शोज दैट द साइज वाज लेस देन 10 नैनोमीटर. तो 10 नैनोमीटर से जब आप साइज रिड्यूस कर लेते हैं, इसकी प्रॉपर्टीज जो है ट्रेमेंडसली इंक्रीज हो जाती है, क्योंकि जो साइज है, जो सरफेस टू वॉल्यूम रेशियो एट दिस लेवल इज एक्सट्रीमली हाई, दैट व्हाट वी वर एबल टू अचीव. फिर हमने जो जब ट्रीट किया माइसिस को, नॉर्मल के हमने हिस्टोपैथोलॉजी की एंड इट वाज ओके. फिर हमने जो वो ग्रुप जो ऑलरेडी इन्फेक्टेड थे, जिसमें कार्बन टेट्राक्लोराइड यूज किया था, and then we also carried out histopathology, and their liver were drastically affected. Then we just treated this with the selumirin, one of the group, and the other we treated with our own selumirin coated gold nanoparticle. And what we observed that a complete recovery of the liver happened when we loaded the selumirin with the gold nanoparticles in the rats. These results are then we patented, and these are some of the results. Since there are students, so I'm not going to discuss the technical details, but then what we observed that we also investigated whether our gold nanoparticle is affecting the other vital organ like kidney, liver, pancreas or heart, but what we observed that these were not affecting the vital organs. So it means that these nanoparticles were safe, because there were at that time when we were working on this, it is like 10 years back, so there were very little information about the toxicology of the nanoparticle. So we investigated its toxicological effect, and what we observed that these nanoparticles were safe, that were not affecting the vital organs, at least at that stage. Similarly, we are also working on the multi-drug resistance bacteria, and you know that the resistance, the bacteria develop resistance against the antibiotic through a number of ways. So I just received message that I should wind up. So these are the ways. I am not going to discuss it in more detail, but what we have done, over the years, we took the commercially available drug and we tried to just reverse the nanoparticle of these drugs and reverse the multi-drug resistance. And then we just investigated them with the help of atomic force microscopy. As you see, this is the E. coli, this is a Strep aureus. So we were able to just analyze the E. coli and the Strep aureus with the help of atomic force microscopy. And then when we treated these microorganisms with our nanoparticles, we were just observing them live through atomic force microscope. And what we observed, I'll just only show results with the cefotriaxone, these are the results that when we treated these E. coli with just 5 milligram for 6 hours, 5 milligrams for 2 hours, these E. coli with the nanoparticle, as you can see that these E. coli were completely disintegrated. And for 8 hours they completely disappears, and these are the results which we were just observing live in the real times, which shows that the nanoparticle just increase the effect of cefotriaxone to a greater extent. And these are some of the other results. And let me thank a wonderful group of my students who worked over the years. My research group, one of my students, Badnawaz, he also worked in this area. So he is assistant professor at Michigan State University. Burhan is also in Canada. Nadeem, Dr. Said Nadeem, he is also assistant professor at Mughla University, Turkey. And thank you so much for your attention. Okay.
Attendees, unfortunately, due to the shortage of time, the question answer sessions will be conducted towards the end of all of the presentations. Thank you very much for that interesting presentation, Professor Dr. Raza Shah. Next, we have Professor Dr. Mohammed Asaf Qureshi. Professor Dr. Mohammed Asaf Qureshi is currently working as a professor of pathology, consultant molecular pathology, director, Dow Labs, focal person, Dow Genomics Lab, and head of the Dow Career Registry at the Dow University of Health Sciences. Professor Qureshi is actively involved in academics, undergraduate and postgrads, research, diagnostic and administrative services at the DUHS. He completed his MBBS from Dow Medical College in 2005, followed by PhD from the University of Glasgow, UK, and postdoc from University Medical Center, Mainz, Germany. Sir, please make your way to the podium.
थैंक यू वेरी मच. गुड आफ्टरनून एवरीवन. कैसे हैं? खैरियत से सब ठीक ठाक? मुझे मेरे कुछ स्टूडेंट्स बैठे हैं ऑडियंस में और मुसलसल मैसेजेस आ रहे थे, सर टॉक छोटी रखिएगा. और मैं उनको ये रिप्लाई कर रहा था, भाई मेरी टॉक शुरू तो होने दें. आप शुरू हुई नहीं और मैं छोटी रख दूं तो द ओनली वे आई कैन डू दैट इज जस्ट गो राइट नाउ एंड से बाय बाय एंड दैट वाज इट. बट आई रियली डोंट वांट टू डू दैट. एनीवेज, जोक्स अपार्ट, आई ट्राई टू बी वेरी क्विक, वेरी ब्रीफ, वेरी क्रिस्प, एंड वुड लाइक टू थ्रो अ मैसेज दैट आई रियली वांट टू थ्रो from this platform. तो सबसे पहले डॉक्टर काशिफ और सारी जो लाइफ साइंसेस की फैकल्टी, थैंक यू वेरी मच फॉर हैविंग मी ऑन स्टेज हियर. एंड आई वुड लाइक टू टेल यू गाइस, जो ऑडियंस में सारे लोग बैठे हैं, कि मेक यूज ऑफ दिस प्लेटफॉर्म. दिस इज प्रोपिशियस फॉर यू, दिस इज इंपॉर्टेंट फॉर यू. ये 15, 20, 25 जो इन्होंने रिसोर्स पर्सन्स आपके लिए अरेंज किए हैं, इंक्लूडिंग माई सेल्फ, डॉक्टर सईद यहाँ है, मुश्ताक है, डॉक्टर रजा, एंड ऑल ऑफ देम, जितने भी प्रेजेंटर्स हैं, दे आर रिसोर्स पर्सन्स फॉर यू. मैं एक दफा लिंडाओ गया जर्मनी, दैट वाज अ नोबेल लॉरिएट मीटिंग. जर्मनी में वो ये एक प्रोसेस करते हैं कि दे इनवाइट यंग साइंटिस्ट्स एंड नोबेल लॉरिएट. आई हैव येट नॉट वन नोबेल प्राइज, बट मैं वहां गया था एज अ यंग साइंटिस्ट. वहां इंटरेक्शन होती है उन सबके साथ. एक स्लोगन रखते हैं वो, एंड द स्लोगन इज कनेक्ट, एजुकेट, इंस्पायर. ये उनका स्लोगन है और मुझे ये स्लोगन बड़ा पसंद है. ऑब्वियसली अगले 5, 6, 8 मिनट में मैं आपको ये पूरी नेक्स्ट जनरेशन सीक्वेंसिंग पढ़ाने नहीं लगा, ना पढ़ा सकता हूं. ऑब्वियसली इट्स अ वेरी लाइक इन डेप्थ टाइप ऑफ टॉपिक, वो हम बाद में बात कर सकते हैं. बट ये मैसेज जो मैं थ्रो करना चाहता हूं, प्लीज कनेक्ट विद पीपल, ट्राई टू एजुकेट योरसेल्फ. Get inspired and inspire other people, okay? My name is Professor Asaf Qureshi and I'm Professor of Pathology at Dow University of Health Sciences. Introduction number थोड़ा उन्होंने दे दिया. I'll be talking about next generation sequencing and very quickly why this is important clinically. और particularly मेरा focus होगा oncology और cancer related NGS पर. Since this is the life science audience, तो next generation sequencing आपके लिए it's a very very important tool for you to know about this. क्योंकि लाइफ में कभी ना कभी आप इसको यूज कर रहे होंगे, so it should be in your skill set list. This is important, guys. Elon Musk का नाम सुना आप लोगों ने? किसने सुना Elon Musk? हाथ उठा के बताएं मुझे. Raise your hands. Right? He's the Tesla guy, right? There is more to it. टेस्ला से ज्यादा आगे भी चीजें हैं. वो रॉकेट्स बना रहे हैं जी, मार्स को कॉलोनाइज करना. You know about this? All of you? Say yes if you know. Say yes. Say नहीं सुनाई दे रहा. A little more loud. Yeah. So you know about. Do you know about Neuralink? You know about Neuralink? This guy has designed microchips which will communicate with the human cortex so that the human and machine interface is improved. Oh, what a guy he is, huh? और सबसे अमीर बंदा भी है और ट्विटर and all these. He's famous these days, isn't he? अच्छा, उससे किसी ने पूछा कि आप लोगों को हायर कैसे करते हैं? How do you hire people? जॉब पे कैसे रखते हैं? कौन सी डिग्री देते हैं? पीएचडी होनी चाहिए, पोस्ट डॉक होना चाहिए. व्हाट शुड द पर्सन हैव? वो जवाब देता है मैं डिग्रीज तो देखता ही नहीं. मैं एक सवाल करता हूं, द फर्स्ट क्वेश्चन, एंड देन द इंटरव्यू टेक्स इट्स ओन वे. और वो सवाल क्या है? यू नो दैट? व्हाट विल यू कंट्रीब्यूट टू माय कंपनी? आप क्या कंट्रीब्यूट करेंगे मेरी कंपनी में? दीज आर माय कंपनीज. आपके स्किल सेट्स, what value will you bring to my company? So please increase your skill set. अपनी values बढ़ाइए, goal set कीजिए, target set कीजिए और उसके लिए फिर आप लग जाएं. और फिर इस पूरे process में जो आप चीज बनेंगे और फिर आप अपने आप को admire करेंगे कि wow. So ये सारी जो workshops होती हैं, educate, connect, inspire. Okay? बात करते हैं next generation sequencing की. मेरे पास ज्यादा टाइम नहीं है. I'll be very quick. और मैं आखिर में आपको बताता हूं कि ये पूरी प्रेजेंटेशन आपको मिल भी जाएगी, आप इसको पढ़ भी लेंगे. मैं अवेलेबल हूं पूरे का पूरा YouTube पर. ये पूरी प्रेजेंटेशन मैंने वहां डाली भी दी है, पढ़ाई भी दी है. So that's not a big deal. पॉइंट सिर्फ इतना है कि there are different range, there's a wide range of genetic variations. और इस स्लाइड का टेक होम मैसेज ये है कि for right diagnostic dilemma you need to pick up the right diagnostic test. अगर घर पे रोटी बनानी हो तो तवा रखेंगे, देग नहीं रखेंगे. देग में रोटी नहीं डालेंगे. So that's the kind of thing that I want to give message इस स्लाइड में कि अगर you are looking for big genetic variations such as chromosomal changes, such as polyploidy, aneuploidy, rearrangements, these are big changes. And these big changes can be obviously detected by something like karyotyping. Easily doable. And as you go down, smaller changes, copy number variants, indels, point mutations, and all these, you need to pick up more relevant, more sensitive technology. NGS is a highly, highly powerful technique, okay? It's out there sequencing for a long, long time now, but a long, long time goes into decades and decades. But anyways, it's making success stories. So NGS अब cheap हो गया है, it is more affordable, it is more available. So ये पूरी timeline है कि जी success story है. पहली oncogene isolate हुई थी somewhere in 1982, that was KRAS oncogene from urothelial cancer. और आज हम बात कर रहे हैं personalized medicine की. That's the other word which I want to pitch out today, personalized medicine. ये NGS का सारा खेल यही है. अगर पूरे लेक्चर को मैं समराइज कर दूं कुछ सेंटेंसेस में तो वो ये है, किसी को कैंसर हुआ, उसका टिश्यू निकाला, उसकी होल जीनोम या होल एक्सोम सीक्वेंसिंग की और हमें पता लग गया वेरिएशंस कौन कौन सी हैं, कौन कौन सी जेनेटिक म्यूटेशंस हैं. सम ऑफ द म्यूटेशंस आर एक्शनेबल. When we say actionable, उसका मतलब यह है कि अगर पता लग गया कि इस पेशेंट में, इस कैंसर में ये म्यूटेशन है, तो ये ड्रग दे दें, काम करेगी. ये ड्रग हर पेशेंट को देने की जरूरत नहीं है, सिर्फ उस पेशेंट को दें जिसमें ये म्यूटेशन है. अगर एक पर्टिकुलर कैंसर में एक पर्टिकुलर म्यूटेशन डिटेक्ट कर ली तो जरूरी नहीं है उसके लिए ड्रग अवेलेबल हो, लेकिन ये हो सकता है कि उससे ये पता लग जाए कि जिन पेशेंट्स में ये म्यूटेशन होती है उनकी प्रोग्नोसिस अच्छी नहीं होती. 5-year survival rate. So the point is कि म्यूटेशन और NGS से नेक्स्ट जनरेशन सीक्वेंसिंग से जो डेटा आपको मिलता है, it may have diagnostic importance, therapeutic importance, prognostic importance. Okay, so this is important. और ये this सक्सेस स्टोरी इज अभी 2022 है और ये बढ़ता जा रहा है. मैं a few months ago I was in US for one of the training sessions और वहां मैं NGS lab में काम कर रहा था and that was a norm there. हर ऑपरेटिव सर्जन वहां पर कैंसर की सर्जरी का सैंपल हिस्टोपैथोलॉजी के लिए भी भेजता है और NGS के लिए भी भेजता है कि जरा देखें ना इसके अंदर कोई म्यूटेशन ऐसी है जो आज की तारीख में एक्शनेबल है और हमें पता है. सो दैट्स अ नॉर्म एंड रूटीन इन वेस्ट many of the hospitals और hopefully पाकिस्तान में भी अभी earlier on यहाँ सेशन था. all these optimist people are here and inshallah we'll bring the change. ये चीजें यहाँ भी स्टार्ट हो जाएंगी बहुत जल्द. and I'll tell you ह्यूमन जीनोम प्रोजेक्ट में इसकी डिटेल्स में नहीं जा रहा. पॉइंट सिर्फ ये है कि सेंगर सीक्वेंसिंग यूज होती थी, आज भी गोल्ड स्टैंडर्ड है, बट दिस थिंग NGS इज मैसिव. इसका थ्रूपुट बहुत ज्यादा है. इट्स मैसिवली पैरेलल सीक्वेंसिंग. आप बहुत जल्दी कर सकते हैं, चीप हो रही है, सो इट्स फास्टर, थ्रूपुट ज्यादा है, बट स्टिल यू हैव टू गो बैक टू सेंगर सीक्वेंसिंग. हैविंग सेड दैट कि जी NGS बहुत तेजी से आगे बढ़ रही है का मतलब ये नहीं है कि सेंगर इज आउटडेटेड, ऐसा बिल्कुल भी नहीं. सो डोंट टेक दिस मैसेज. ओके, NGS के बहुत सारी मशीन्स अवेलेबल है. इल्यूमिना बाय फार इज द मार्केट लीडर. वो कहते हैं कि मोर देन 90% ऑफ द सीक्वेंसेस जो कहीं पर भी दुनिया में सीक्वेंस हो रही हैं वो Illumina से है. So Illumina is the market leader, but there are so many other platforms you should know about sequencing. These are babies from Illumina, MySeq, HiSeq and NovaSeq. We are also establishing a genetics lab at Dow University of Health Sciences, almost done, will be inaugurated I think in a couple of months time. We will invite you guys there to have a look at how these machines look like और क्या workflows होते हैं. Okay, so we are getting a couple of MySeq and we are also getting a NextSeq. So these are the machines which do the sequencing and the reaction. जैसे ELISA करते हैं, PCR करते हैं, 96 well plates होती हैं, इनका जो reaction का platform है वो कहलाता है flow cell. It's a flow cell. Okay, इस डिटेल्स में अभी नहीं जाते. ये वर्ड सुन लो जरा, समझ लो कि ये इंपॉर्टेंट है. हम कहते हैं कि मैसिवली पैरेलल सीक्वेंसिंग सेल्फ एक्सप्लेनेटरी है. मैसिवली बहुत ज्यादा पैरेलल सीक्वेंसिंग. तो जो PCR रिएक्शन यहां हो रहा होता है, बिकॉज दिस इज सीक्वेंसिंग बाय सिंथेसिस, वो PCR रिएक्शन एक ही पॉइंट का बहुत सारी जगहों पे हो रहा होता है. ये सारा इट्स गोइंग ऑन इन पैरेलल एंड दिस इज द रीजन इसका थ्रूपुट बहुत ज्यादा है. ओके, टेक्निकल डिटेल्स में इसमें भी नहीं जाता कैसे करते हैं, जस्ट फॉरगेट अबाउट इट. ये स्लाइड इंपॉर्टेंट है. हम में से बहुत से लोग लैब में काम कर रहे होते हैं, लाइफ साइंसेस के लोग भी हैं, पैथोलॉजिस्ट हैं, वर्किंग इन द लैब. बट ये जो ब्रिज है ना कि सैंपल हमारे पास आता है और क्लिनिक में बैठा हुआ डॉक्टर आपकी रिपोर्ट का इंतजार कर रहा होता है कि उस रिपोर्ट में हम लिखते क्या है. दिस इज हाउ मेडिसिन वर्क गाइस. मेडिसिन ऐसी काम करती है ना, पेशेंट आपके पास आता है विद सम प्रेजेंटिंग कंप्लेंट्स, डॉक्टर उसकी हिस्ट्री लेता है, उसकी एग्जामिनेशन करता है, फिर एक लेबोरेटरी इन्वेस्टिगेशन करवाने के लिए उसको लिख के देता है, ब्लड CBC कराएं, LFTs कराएं. सो ये जो लैब वाला बिट है ये बड़ा इंपॉर्टेंट है, क्योंकि सैंपल जो कि क्लिनिक से आपके पास आया है और इसकी रिपोर्ट वापस क्लिनिक में जाएगी, इस बीच में जितना कुछ है, दिस इज ऑल द लेबोरेटरी इन्वेस्टिगेशन एंड दिस इज वेयर यू एंड आई आर वेरी इंपॉर्टेंट. ओके, सो ये सैंपल जो हमारे पास आता है बेसिकली हम उससे DNA एक्सट्रैक्ट करते हैं. ऑल्सो RNA, इफ यू आर डूइंग RNA-seq, वी डू लाइब्रेरी प्रिपरेशन इज अ स्टेप फॉर डूइंग सो. सीक्वेंसिंग करते हैं, देयर इज अ लॉट ऑफ बायोइन्फॉर्मेटिक्स इन्वॉल्व्ड एंड देन वी रिपोर्ट ऑब्वियसली. और ये रिपोर्ट फिर क्लिनिक्स में डॉक्टर्स पढ़ रहे होते हैं कि अच्छा ये म्यूटेशन है, तो इस म्यूटेशन का मतलब क्या है, मुझे ये दवाई देनी है नहीं. बिल्कुल आसान करके अगर समझाऊं, ब्रेस्ट कैंसर सबको पता है, इज द मोस्ट कॉमन कैंसर इन पाकिस्तान. ब्रेस्ट कैंसर में एक ड्रग यूज की जाती है दैट इज कॉल्ड हर्सेप्टिन. हर्सेप्टिन इज अ वेरी एक्सपेंसिव ड्रग एंड इज नॉट फ्री फ्रॉम साइड इफेक्ट्स. उसके सीरियस साइड इफेक्ट्स हैं और बहुत एक्सपेंसिव है. हर्सेप्टिन सिर्फ उस ब्रेस्ट कैंसर के पेशेंट को देते हैं जिसमें HER2 एक प्रोटीन एक्सप्रेस होती है. यानी इट इज इंपॉर्टेंट टू नो कि अगर ब्रेस्ट कैंसर का पेशेंट है, ब्लाइंडली मैं सबको लिख के नहीं देता जी हर्सेप्टिन ले लें जी हर्सेप्टिन, ऐसा नहीं है. ब्रेस्ट कैंसर, देन आई वुड लाइक टू नो HER2 एक्सप्रेस हो रही है या नहीं. अगर HER2 एक्सप्रेस हो रही है तो हर्सेप्टिन देंगे. पर्सनलाइज्ड मेडिसिन सिर्फ उसको देंगे जिसमें HER2 एक्सप्रेस हो रही है. Okay, so that's what the sort of NGS report. Lung cancer, there are around 500 actionable mutations known to date. Around 500 actionable mutations. और actionable mutations 500 इस वक्त पता है, या उनका कोई ना कोई therapeutic implication है, या diagnostic role है, या उसका कोई prognostic role है. Okay, so that's pretty important stuff to know. आपको पता होना चाहिए NGS है क्या, क्लिनिक्स में उसका क्या यूज है, पाकिस्तान में कहां कहां हो रहा है. देयर आर अराउंड 13-14 साइट्स इन पाकिस्तान जहां इल्यूमिना की मशीन्स रखी हुई हैं और काम हो रहा है. Now Dow is also going to inaugurate one of the centers, so you should know कि कहाँ sequencing हो रही है. अभी मुश्ताक अपनी टॉक में बता रहे थे they did sequencing, I'm sure या उन्होंने यहाँ से कराई होगी या कहीं भेजी होगी बाहर. So you should know कि resources क्या हैं, कहाँ से हमको sequencing करवानी है, right? Knowing your sample is important, नहीं करते इस पे बात. जैसे जैसे चीजें इवॉल्व करती हैं डायग्नोस्टिक मेडिसिन में, चीजें आसान होती जाती हैं, they become easy. अब पैनल्स आते हैं कि जी एक पैनल ये लगा दें, फॉर एग्जांपल अवेलेबल किट है फाउंडेशन 1, ये क्या करता है जी, ये 324 जीन्स को टेस्ट करके बता देता है कि उन 324 जीन्स में से कौन सी म्यूटेटेड है. ऑब्वियसली ये 324 वो जीन्स होंगी जो किसी ना किसी कैंसर में कोई ना कोई रोल प्ले कर रही होंगी, इनके लिए कोई ना कोई एक्शनेबिलिटी अवेलेबल होगी. सो दीज पैनल्स आर अवेलेबल इन मार्केट, देयर आर मेनी मोर देन दीज, ओके. एंड दिस इज द ब्यूटी, वो कहते हैं ना ये रिसर्च के बाद और जो सक्सेस स्टोरी का जो मजा है वो यहां है इस स्लाइड में. पर्सनलाइज्ड मेडिसिन, हाउ, मतलब मुझे तो रात को ख्वाब में भी आती है ये स्लाइड, आई होप आपको भी आए. जरा देखिए ना इसका मैसेज क्या है, मैसेज सुने. यानी अगर किसी को लंग कैंसर है और उसमें EGFR की म्यूटेशन है तो बॉस ये दवाइयां दे दो, पेशेंट के लिए फायदा है. हम यहां तक पहुंच गए हैं, दिस इज अ बिग न्यूज. एटलीस्ट इसके लिए क्लैपिंग कर लो यार प्लीज. यहां तक पहुंच गए. यानी एक टाइम वो था मलेरिया का कुछ पता नहीं था करना क्या है, और आज इतना स्पेसिफिक पता है कि अगर किसी को लंग कैंसर में म्यूटेशन ये वाली नहीं लेकिन रीअरेंजमेंट है इन जीन्स की, तो फिर ये वाली ड्रग देनी, कोई और ड्रग नहीं देनी. दिस इज बिग, इट हैज बिग इंपैक्ट. एंड इमेजिन वेयर वी आर मूविंग टुवर्ड्स पर्सनलाइज्ड मेडिसिन. किस पेशेंट को क्या दवाई देनी है, कैंसर की मैनेजमेंट. This is high yield stuff, okay? So personalized medicine, this is an important slide for you to remember. Obviously NGS reporting is evolving, database emerge हो रहे हैं, ज़्यादा से ज़्यादा लोग ज़्यादा से ज़्यादा genes report कर रहे हैं, उनमें genetic variations report कर रहे हैं, उनकी actionability report कर रहे हैं, और ये data बढ़ता, जितना data बढ़ेगा उतनी एक्शनेबिलिटी इंक्रीज होगी, ओके. सो दिस इज ऑब्वियसली सॉफ्टवेयर्स आर इमर्जिंग, बायोइन्फॉर्मेटिशियंस उसमें अपना रोल प्ले कर रहे हैं. सो दिस इज ऑब्वियसली एन इवॉल्विंग फील्ड, और जो कोई इवॉल्विंग फील्ड होती है वो कहते हैं बैठी गंगा में हाथ जरूर होना चाहिए. दिस इज इन गाइस. स्कॉलरशिप की आप बात कर रहे थे, प्रोजेक्ट बनाइए अराउंड द लेटेस्ट टेक्नोलॉजीज, भेजिए पूरी दुनिया में लोगों को कि मैं इस पे काम करना चाहता हूं, देखें कैसे इजीली स्कॉलरशिप मिलती है. तो somebody saying there obviously कि मुश्किल है, ऑब्वियसली मुश्किल है, बट इसका सक्सेस रेट यूं है. मैं भी जब स्कॉलरशिप के लिए अप्लाई करता था, आई गॉट PhD फ्रॉम ग्लासगो स्कॉलरशिप, पोस्ट डॉक फ्रॉम जर्मनी, जर्मन स्कॉलरशिप. स्कॉलरशिप्स आर अवेलेबल, लेकिन 100 लोगों को लिखें और एक्सपेक्ट करेंगे 2-3 लोग जवाब देंगे. तो जब मुझे लोग कहते हैं ना सर कोई जवाब ही नहीं देता, तो मेरा अगला सवाल होता है 100 लोगों को लिखा, 100 emails, did you send out 100 emails? नहीं सर 100, तो आपके ख्याल में क्या मतलब, आप फारिग बैठे हैं, आपने ईमेल की, आपको बोले आ जाए जी आ जाए, ऐसा नहीं है. It's not like this. So you have to work hard if you want to achieve something, okay? So that's the take home message. This is simply conclusive remarks. Obviously we need to pay attention. NGS is a new thing. इसकी अवेयरनेस बढ़ानी होगी क्लिनिक्स में भी, और जाहिर पाकिस्तानी पर्सपेक्टिव इज डिफिकल्ट पर्सपेक्टिव. यहां मैं जब जाता हूं ऑन्कोलॉजिस्ट से बात करता हूं, क्लिनिशियंस के साथ बैठता हूं कि जी ये एक चीज है जो हम पाकिस्तान में इंट्रोड्यूस करने लगे हैं डाउ यूनिवर्सिटी के प्लेटफॉर्म से. अरे यार डॉक्टर साहब यहाँ पेशेंट 300 रुपए का टेस्ट नहीं करवाता, ये कितने का होगा? मैंने कहा यार ये तो हजारों का होगा, 30, 40, 50 हजार का टेस्ट. अरे कहां करवाएंगे? दीज आर द रियलिटीज जो हमें फेस करनी है, बट दैट डजंट मीन कि हम ये काम ना करें. So we have to know कि जी क्या ground problems हैं, और उसमें रहते हुए we have to find the solutions, okay? Cost still is a major challenge. I think we should not have more facilities because these equipment are very expensive. 8-8 करोड़ का एक equipment है, और हमारे पास पाकिस्तान में 13-14 जगहों पर रखा हुआ है, और 14-15 की जगह हमारी है डाउ की. मेरे ख्याल है दिस हैज टू स्टॉप. सेंट्रलाइज्ड फैसिलिटीज हो प्रोविंशियल लेवल्स पे, और सब उस फैसिलिटी को यूज करें, इज द सॉल्यूशन, और इससे कॉस्ट भी कम होगी ऑब्वियसली. पर्सनलाइज्ड मेडिसिन रिक्वायर NGS टेस्टिंग, वेरी इंपॉर्टेंट. और ये पर्सनलाइज्ड मेडिसिन का कॉन्सेप्ट लेके जाना है आज यहाँ से स्टूडेंट्स. अवेयरनेस बढ़ानी है, नॉट ओनली इन द क्लिनिशियंस बट ऑल्सो इन द पेशेंट्स, ओके. Or NGS testing is evolving. So that's all from my side. I don't want to bore you with the techniques of NGS. लेकिन if you are interested to know more about it, so आप connect हो जाए मेरे साथ. इसका ये डिटेल लेक्चर मेरे चैनल पे पड़ा हुआ भी है. Just type in Asaf Qureshi at YouTube. You should be on my channel. And if you have any problem and if you want to connect to me, this is me. Just text me. Okay. So one thing you should do, because we do not have time, is to save this slide if you want to get back to me. Okay. Thank you very much. If you have any questions, I can take them now or later, or whatever the management decides. Later? Okay. But you should at least appreciate I was the quickest of the speakers. Yeah.
Thank you very much for that very interesting presentation. It was precise but very thorough. Next, last but certainly not least, we have Dr. Sikandar Azam. Dr. Sikandar has research experience particularly in computational chemistry and molecular dynamic simulations of protein complexes to understand their structural and dynamical aspects and role in biological systems. Dr. Sikandar accomplished his PhD from the University of Innsbruck, Austria in 2009 and had postdoctoral experience in the US and Europe. Furthermore, he is a member of the Pakistan Academy of Sciences and IWAS Young Affiliate who established computational biology at the National Center for Bioinformatics, Quaid-e-Azam University, where he is serving an associate professor. Associate Professor. Dr. Sikandar has achieved 5 national and 5 international awards. Ernst Mach Follow-up Award has recently been awarded to him by the Austrian Science Foundation. He is the reviewer and editorial board member of many international journals. Recently, one of his researches on COVID-19 was published in PLOS 1 and European Journal of Pharmaceutical Sciences, and he will discuss some of his findings with us. Dr. Sikandar, please make your way to the stage and present your findings.
पॉकेट में रख लेता हूँ. इससे नेक्स्ट पर ऐसे.
Well, good afternoon everyone. Can you hear me? So my task is very difficult and the topic of my talk is how to make you not sleep right now because it's end of the day. Thank you for the nice introduction and thank you for Professor Dr. Kashif and his team and Dr. Hamad Kiani and everyone who has given me opportunity to speak. So trying to, okay, well, where I'm from Computational Biology Lab from the National Center for Bioinformatics, and this is the area I have been working on the molecular dynamics simulation of proteins since last many years. So computational biology is actually the, well, it's an amalgam of various disciplines which is actually, you need the information of genomics, proteomics, and you need the information of molecular biology, and certainly you will collect all those information and combine it with the computer interface, and then you know what is computational biology. So this is the opening area of bioinformatics where graduates and undergraduates are highly in demand. So I will skip those things which already have been discussed. So this will be the outline of my talk. So as you know, the computer speed and capacity have paved a new way, opening new horizons, new disciplines. So just same like there was a time when people were sending their letters in hard form, now you have, of course, email, Facebook and all that. So the computational biology has actually revolutionized all other fields as well, which are mentioned here. So starting with I will skip this. Okay, this will be the major slide. Let's go back to FSC and we will talk about the molecular dynamics now. In molecular dynamics, when you are treating with proteins, everything is a molecule. So you have to take them as a molecule. So if you remember, if I am standing here, so I am just addressing the students and I'm asking them this question. if I am here and I change my position to this place, so what I have done? So this is the only way not to make you sleep. I have covered what? Distance. And if this is in a particular direction, what is this? Displacement. Excellent. So I am changing the position from x1 to x2, right? And I am, when I include time in it, what the term we use? Movement, motion, right? And now you all know the laws of motion. So this was the laws of motion Dr. Zuhair Heather was talking about. These things are governing these all discoveries. So you treat the molecule as one, and then you make them in the motion, and then you just inquire what's happening inside. So let's make up in mind there is a disease you want to work on. This will be the last talk, and I want to give you some information that how to start. Okay, just what I usually do in my class. How to start? Let's think about any disease in your mind. What is the journey? Come on. The journey is like, this is the traditional journey I'm talking about. So a drug, you identify the disease, you go for the isolation of protein, you find the drug effective for it, you take 5 years, preclinical testing, formulation. These are those steps which makes a drug to come into market in 15 years. So previously it was like that. So what happens after the recent trends of genomics, proteomics, bioinformatics, and eruption of computer-aided drug designing methods? So what happened? The duration which was actually required for the previous traditional method has been reduced to a large extent, right? So I am not going into the detail of these techniques. This is high throughput screening. What we are doing, we are doing as our work. So this is the main combinatorial chemistry. You see, it's a journey of having a lot of compounds. We started with the 9 lakh compounds, right? With 9 lakh compounds, we filtered 7 lakh compounds. And after that 7 lakh compounds, we came back with the computational methods to 100 compounds. These 100 compounds were further tested, and we came up with 74 compounds. So I'm not very much overwhelmed, but scientists are unable to circumvent the problem of antibiotic resistance because we don't have much antibiotics. We don't have options. So cutting this into short, this is the combinatorial chemistry which I want to talk about, but okay, I will give you the hint that, did you remember the carbon chains? Right? The 3 carbon chain is propane. And if there is a Cl attached, it is chloro. So just imagine in your mind that the property of just propane and chloropropane will be different, right? Okay, so these are the possibilities which we play with the functional groups, and we have enormous amount of softwares through which we can screen millions of compounds, and there are jobs in Europe, in US for you people. You know, if you are good screener and you can screen, when there is a capacity, okay, he can screen 2 million, 3 million, 4 million, it's something like that. So going ahead, molecular modeling, this is another domain of bioinformatics and molecular dynamics. When the modeling was improved, it's not modeling, it's molecular modeling. So it's different from that modeling. So in modeling, you have a model who is an ideal, right? And he or she walks on the ramp. And molecule, you have to select a molecule which is ideal to be a drug. And when you watch it in the software, this is called molecular modeling. So I related. Well, 10 to 15 years, and there is a huge cost. Just Dr. Bal Chaudhary also mentioned that there is billions of rupees which is required. Okay, now I am trying to solve your problem. You people are tired? Yeah. So here is your. So when you are tired, when you are fatigued, you are like having headache, fever, something should come into mind. Here are just numbers. I have shown this to slide. This number is actually, we are just numbers, right? But this is the number 1877, when there was a person who actually synthesized a drug, right? In 1893, it was first used by von Neumann to the human. From 1893 to till 1950, it was always controversial. Oh, there are safety problems. Might be it comes into the market. No. There are this problem, that problem. In 60s and 70s, ultimately there are some companies, they say, okay, just market it. This is the solution of your headache. Before that, people were taking just salicylic acid. This is the journey of Panadol, which you are taking for good for nothing. Oh, I'm tired, I just one pill and I'm okay. Right? So what I'm trying to tell, that every subject has a story. Every drug, every medicine has a story. Well, there are some facts which Dr. Mushtaq has really explained a lot, so I will skip that part. So we actually developed some constructs on the COVID for the design, and it was the first paper from Pakistan which was published from our group, and which came into the media. We just developed 5 constructs, and it was published in European Journal of Pharmaceutical Sciences. And the second part is the multi-drug resistance and introduction, which I am totally skipping. This is again, you go to the doctor and you are resistant. We are talking about it a lot. So this is because, don't put everything on the doctor. How much of you know your own medical allergies? When doctor asks you, do you have any medical allergies? No, I am fit, I am Superman, I am Captain Marvel. You don't know about yourself, but you blame the doctors. Because I have been in contact with doctors as well. It's not like that there are some extent, there are some doctors who are giving you the over-the-counter antibiotics and making you resistant, but it's not all their fault, right? We have overburdened doctors, 10,000 people and one doctor. So you have to know about yourself as well. If you have viral, come on, listen to me this carefully, the take-home message. If you have a viral which should go by 2 or 3 days, don't take any medicine, right? You feel it, okay, this is some allergy, don't go for the extra medicines on it. Okay, I'm skipping this because these are introductions, and then I will come. Okay, this is our route. Yeah, in 5 minutes I will be done. This is our route. In bioinformatics we have developed some methods, which is, of course, we have written for the patent and the copyrights as well. So what we do the computational part, then experimental part we collaborate, and we also do the algorithm designing. This was another discovery of a novel beta-lactamase inhibitor. This was the journey which I am talking about, that 7 lakh, and then we have 74 compounds. And after those 74 compounds, we came to know that beta-lactamase is like making a lot of mess inside. So we came up with the inhibitor after in collaboration with US in Maryland. So we came up with one single compound after that journey, and that compound was very effective. So what is antibiotic resistance? That you already know, right? That this antibiotic, when binds, it's not functioning, right? So what should we do? Okay, let's see, this is a bottle. Okay, I can't go from this side, and I'm in desert, there is no water, I am in Sahara. I have this pointed nib with me. How can I, if I want to drink water, what I will do? Yes, thank you. I will make a hole. So our compound is doing that. We are giving it for combination therapy, I don't know means I am unable to explain all that paper. You can go for the PLOS one, this paper, and you can watch the details. So this is the thing which our compound is doing. So this was a journey of 3 and 4 years through. We worked with the silks, with the charm person, Alexander Mackerel, and Adrian Mulholland. So we have some collaborations in UK and US. So this is what actually was happening. With our inhibitor, it was, without our inhibitor, it was everywhere in the resistant, in the clinical strain, in everywhere it was doing that function, and it was okay. But when we applied the inhibitor, it was stopping the beta lactamase to be. So we checked it in recent cutting edge technology of molecular fragmentation, which is called silks. It's not mere docking, it's not mere dynamics. It's the dynamics which has been an amalgam of charm and amber. So I will not go into the detail. There are respective sites which are actually working. So we found some respective sites. Then after side by side we checking it in the experiments, and we found good values for compound number 11, which we have filed. So when we check in the bacterial cell wall, inside the cell wall the penetration was very good for that compound. So well, I will not go into the detail of this article, except that I have presented a theory here which is called theory of retaliation, that we all should be responsible for what we are having, for a balanced diet, for a better balance of ecosystem. Not always, don't go for the meat because it is making an imbalance. You should have a balanced consumption. So this theory of retaliation is there, that we are consuming the bats and all the animals, and they are retaliating it. So if anyone is interested, I can give this paper to you. You can read it out and you can know what is the exact theory of retaliation is. What I am trying to tell is that that the overconsumption of these food and unbalanced consumption is actually lethal. So last but not least, this is my research at a glance, in which my group is working. I have produced 60 MPhil students with 5 PhD students. These are our methods which we actually developed. For one is indigenous method with my student. So if you say there is no job, my student has completed his PhD in December. In April, he was having H1 visa for US and a job, and his salary was actually more than my salary, and he was in industry in Maryland. So it's not that job will come to you, it's you will make the job. It's like water is running itself and making a way. Okay, so these are some glimpses of the method which we developed, and this is another. Okay, this is my group. These are our activities, and these are my collaborators. I am very thankful to them. Thankful to the President SZABIST, Dr. Shahnaz Wazir Ali, and Dr. Kashif, and thank you for your patience. Well, I am open to questions if there is any.
Attendees, please give a huge round of applause for our guest speakers, Dr. Mushtaq Hussain, Dr. Raza Shah, Dr. Asaf Qureshi, and Dr. Sikandar. To show our appreciation, I would like to invite Dr. Hamad Afzal Kiani onto the stage to present the guest speakers with shields. honorable guests, as you hear your name, please make your way towards the stage and receive your shields. Professor Dr. Mushtaq Hussain. Professor Dr. Raza Shah. Thank you. Professor Dr. Mohammed Asaf Qureshi. And last but not least, Professor Dr. Sikandar Azam.
I would now like Dr. Hamad to present the shield to our Master of Ceremony today, Anusha Khan, as a token of appreciation.
Thank you all so much for being part of the SZABIST first international conference, LIFSCICON 2022. The conference will continue tomorrow with a lot more exciting knowledge in store for all of us. With this, I, Anusha Khan, bid you all a farewell. Enjoy the rest of the evening. Thank you.
ये जगह सही है लगाने की। चलो आ जाओ भाई, जल्दी आओ। क्या वो मोबाइल इशू है? नहीं सर, मोबाइल है।
आओ, मोबाइल दे दो किसी को
रिक्वेस्ट। ये भी हमारी क्लास का हिस्सा है ना।